Infectious diseases, preventive medicine and clinical species conferences too. Brucellosis is caused by facultative intracellular alpha proteobacteria of the genus Brucella, which is related phylogenetically to other bacterial genera such as Agrobacterium, Phylobacterium, Rhizobium, and Acrobactrum. Bacteria of the genus Brucella are gram-negative cocci, coccobacilli or short rods, with straight or slightly convex sides and rounded ends, that do not ferment carbohydrates in conventional media. They are transmissible to a wide range of animal species. Early bacteremia is followed by localization of the infection in the genital organs and cells of the monocyte-macrophage series. Brucellosis is an important disease of humans, domestic and wild animals worldwide. In humans, the disease caused by Brucella spp, mainly Brucella melitensis, abortus and suis, is also known as Malta fever, Mediterranean fever or Bang's disease, or as undulant fever. Among domestic animals, cattle, sheep, goats and pigs are mainly affected. Table 1. A number of wildlife species, such as African buffalo, antelope, bison, camels, elk, reindeer, caribou and other wild ruminants and carnivores, are also susceptible to infection. Brucella species and preferential livestock species. The genus Brucella contains many species that include B. abortus, B. melitensis, B. suis, B. ovis, B. canis and B. neotomae. 7 are recognized for B. abortus, 3 for B. melitensis and 5 for B. suis. However, the degree of genetic relatedness, as shown by DNA hybridization studies and whole genome sequencing techniques, is consistent with the existence of a single species within the genus Brucella. Brucella abortus, B. melitensis, B. suis and B. neotomae generally occur in the smooth form, while B. ovis and B. canis are invariably rough species. Recently new Brucella species have been described besides the 6 classical species, Brucella seti and Pinnipedalis in cetaceans and pinnipeds, respectively, Brucella microti in vole and soil, Brucella vulpis in red foxes, Brucella inopinata in frogs, and Brucella papionis in baboon. Clinical manifestations are abortion, the birth of weak offspring, orchitis and epididymitis. Signs of arthritis may be evident in chronic infections. The primary sources of contamination of the environment are fetal membranes, fluids and vaginal discharges expelled by infected females when they abort or at parturition. Brucellae are also commonly shed in milk and semen. The rare occurrence of brucellosis in domestic animals in developed countries is directly linked to the success of brucellosis control/eradication programs. In cattle, biovars of Brucella abortus are usually the cause of brucellosis, but in some countries, particularly in southern Europe, Central Asia and in the Middle East, Brucella melitensis has also been implicated as a cause of abortion in cattle where they are kept in close association with infected sheep or goats. Occasionally Brucella suis may infect the mammary gland of cattle, but it has not been reported to cause abortion in this species. Caprine and ovine brucellosis, excluding Brucella ovis infections, are most commonly caused by Brucella melitensis, but sporadic cases are caused by Brucella abortus. In sheep and goats, the disease caused by Brucella melitensis and its epidemiology are very similar to those of Brucella abortus infection in cattle. Importantly, in West Africa there is no report of the isolation of Brucella melitensis in sheep and goats, and infection in small ruminants seem to be due to Brucella abortus. Brucellosis in pigs is caused by Brucella suis and is characterized by an initial bacteremia followed by the production of chronic lesions in the bones and reproductive organs of both sexes. Brucella suis consists of 5 biovars. Pigs are infected by Brucella suis biovars 1, 2 or 3. Disease caused by biovars 1 and 3 is similar, while that caused by biovar 2 differs in its pathology, host range, and geographical distribution, mainly in Europe. Brucella suis biovar 2 is rarely pathogenic to humans, whereas biovars 1 and 3 are highly pathogenic and cause severe disease in humans. Brucella ovis is one of the most common causes of epididymitis in rams but is a rare cause of abortion in ewes and neonatal mortality in lambs. Low reproductive rates may occur in affected flocks. It is not a zoonosis. Brucella canis causes epididymitis and orchitis in male dogs and metritis in bitches, and is a rare infection in humans. It does not infect other animal species. Brucella neotame has been isolated from a rodent species in western USA. The presence of Brucella spp in abortion material or vaginal discharge can be demonstrated microscopically in tissues or smears using modified acid-fast staining, stamp staining, and is considered presumptive. Polymerase chain reaction, PCR, methods are nowadays used routinely in diagnostic laboratories to identify Brucella DNA in clinical samples. The first species-specific multiplex PCR assay for the differentiation of Brucella, named Amos PCR, on the basis of the initials of the species that it identifies, was described in 1994. This assay was later updated to identify B. abortus vaccine strains S19 and RB51. A real-time PCR was developed in a multiplex format in 2004, allowing the rapid identification of Brucella spp, abortus, and melitensis in a single test. Brucella spp should be isolated by culturing samples on selective media, feral, allowing the further isolation of DNA to perform molecular typing by methods such as multiple locus variable number tandem repeat analysis, MLVA, and multi-locus sequence typing, MLST, as well as whole genome sequencing, WGS. However, PCR-based typing methods as well as WGS become important tools for studies related to the phylogeny, taxonomy, epidemiology and evolution of Brucella spp. Species and biovars identification by classical biotyping methods remains the reference method. Most of the serological tests for the diagnosis of smooth Brucella spp infections have been developed to detect antibodies directed against antigens associated with the smooth lipopolysaccharide and are shared by all the naturally occurring biovars of abortus, melitensis and suis. Diagnostic antigens must be prepared from smooth strains of abortus, or melitensis and comply with minimum standards for purity, sensitivity and specificity. Such antigens are suitable for the detection of antibodies directed against melitensis, abortus and suis in different animal species infected with smooth Brucella. Because the SLPs is absent in the naturally rough ovis and canis, specific ovis and canis antigens, mainly are epitopes, associated with the rough lipopolysaccharide, RLPs, have to be used for the diagnosis of infections caused by these organisms. The Brucellosis allergic skin test can be used as a screening or complementary test in unvaccinated animals, provided that a purified lipopolysaccharide-free, standardized antigen preparation, consisting mainly of Brucella cytoplasmic proteins, is used. It is recommended by the World Organization for Animal Health, OIE, that sheep and goats should be immunized with B. melitensis strain Rev.1 live vaccine against both Brucella melitensis and Brucella ovis infections. In cattle should be used Brucella abortus strain 19 live vaccine and strain RB51 live vaccine, produced from a laboratory-derived rough mutant of smooth Brucella abortus strain 2308. No vaccine is recommended by the WA for use in pigs. Vaccines for the prevention of brucellosis have to fulfill minimal requirements and their production should be based on a seedlot system. Seed cultures to be used for vaccines should originate from reference centers and they must conform to minimum standards for viability, smoothness, residual infectivity and immunogenicity. The World Health Organization, WHO, Laboratory Biosafety Manual classifies Brucella in risk group 3. Bovine brucellosis, contagious abortion by Brucella abortus infection. Bovine brucellosis is a highly contagious disease caused mostly by Brucella abortus, a bacterium which occurs intracellularly in its mammalian host. Apart from causing characteristic mid to late-term abortion and infertility in cows, Brucella abortus also occasionally causes orchitis and inflammation of the accessory sex glands in bulls. Other livestock and wild animal species, though of varying susceptibility, are sometimes infected. Bovine brucellosis is also an important zoonosis. In some countries, particularly in southern Europe and Western Asia, where cattle are kept in close association with sheep or goats, infection and abortion can also be caused by Brucella melitensis. Occasionally, Brucella suis may cause an infection in cattle but has not been reported to cause abortion. The World Organization for Animal Health website and the WAHIS interface, information on the worldwide brucellosis status as well as those animal diseases listed by the WA due to their implications for international trade or public health can be obtained. Brucella abortus is a small, gram-negative, non-sporulating, non-encapsulated coccus, coccobacillus or short rod, 0,6 to 1,5 microns in length and 0,5 to 0,7 microns in width. The organism is not acid-fast but does resist decolorization by weak acids and thus stains red with Stamps modification of the Ziehl-Neelsen stain. Most wild strains are fastidious and slow-growing, and require carbon dioxide, 5 to 10%, supplementation for primary isolation at an optimal growth temperature of 36 to 38 degrees Celsius. Brucella abortus strain 19 is an attenuated strain of reduced virulence which is used for the production of a live vaccine. It grows well in a normal atmosphere at 37 degrees Celsius. Complex media, containing serum, are required for the growth of B. abortus and, although most strains grow on sheep blood agar, the colonies may not be as distinctive as when grown on serum dextrose agar. Growth, on primary isolation, is seldom clearly visible before 48 hours of incubation, at which stage the colonies are usually 0,5 to 1. 0 mm in diameter. The use of selective media, such as Farrell's medium, may substantially enhance the chances of isolation by inhibiting the growth of contaminants. The growth rate of B. abortus may, however, be markedly retarded by selective media and for this reason such cultures should be incubated for 5 days or longer. Smooth colonies on a clear growth medium, such as serum dextrose agar, are convex, entire-edged, have a smooth, shiny surface, and are pale yellowish-brown when viewed under transmitted light. Considerable variation of color and surface texture are found in the rough strains. Smooth forms are often markedly pathogenic whereas the rough variants are usually less so. There is no single test by which B. abortus may be identified with absolute certainty, but a combination of growth characteristics, colonial and cellular morphology, staining properties, agglutinating antisera and biochemical reactions will allow an accurate identification. 8 biovars are recognized, which may be differentiated by phage typing, monospecific antisera, biochemical reactions and growth inhibition tests. There are no proven differences in the pathogenicity of field strain biovars. Several molecular-based protocols have been developed for identification of Brucella spp. To the genus, species and biovar level. Bovine brucellosis is dynamic disease, with emergence of new areas of infection and re-emergence of infection in areas where infection existed earlier. Some countries in the developed world are declared officially free from bovine brucellosis after implementing costly control and eradication programs for decades. In Latin America, brucellosis is one of the most important zoonosis because of its impact on public and animal health, and on the economy. Its distribution is closely associated with the distribution and density of livestock. Data extracted from a small number of quality-assured studies indicate that brucellosis is endemic at high levels in both small and large ruminants in some Middle Eastern countries, such as Egypt and Jordan. India is the world's leading milk producer, contributing around 17% of the world's total milk production and has the world's largest dairy cattle population at around 300 million. In 2019, the overall true prevalence of brucellosis observed in cattle was 8.3% and in water buffalo 3.6%. The highest prevalence of brucellosis was reported in the state of Punjab in Pakistan in both cattle and buffalo. A recent systematic review indicated that the overall seroprevalence of brucellosis in dairy cattle herds in China was 1.9% during the period 2008 to 2018. In northern China, where the traditional agro-pastoral areas and the main animal breeding industry are located, the seroprevalence was greater than 10%, whereas it reached only 5.5% in southern China. No accurate figures are available on the prevalence of brucellosis in cattle in Africa, as most reports are based on non-representative laboratory results. A series of cross-sectional studies carried out in peri-urban dairy production areas of 7 countries in West and Central Africa have recently provided much-needed prevalence estimates in these expanding production systems. The results of these studies confirm that Brucella spp circulates among peri-urban dairy farms in the region, with more than 1/3 of the herds in some areas such as peri-urban presenting evidence of infection. Interestingly, the studies provide some evidence suggesting that intensive systems that are gradually replacing the traditional practice of transhumance and in which cattle are kept confined all year round may increase the risk of infection. Mixed farming, and especially keeping small ruminants along with cattle, a common practice in low and middle income countries, is a recognized risk factor. Cattle usually become infected after ingesting contaminated feed or water or licking an infected placenta, calf or fetus, or the genitalia of an infected cow soon after it has aborted or calved, at which time very large numbers of Brucella abortus are present, particularly in the placenta lochia. Animals may also become infected by inhaling organisms or through the conjunctiva. Calves may acquire infections in utero, or they may become infected after ingesting infected colostrum or milk. Although most will rid themselves of the infection within a few months, a low percentage may remain infected for life and may spread the disease at their first and subsequent parturitions. Infected animals usually abort only once, and subsequent calves are carried to full term although they may be infected. Approximately 2.5 to 9% of heifers born of seropositive cows may be latently infected but serologically negative until the middle of their first gestation or even later, when, for the first time, antibodies to B. abortus may be detectable or abortion may occur. Bulls are more resistant to B. abortus than heifers and cows. Bulls may become infected in utero or during early calfhood by the oral route and retain the infection into adult life. In bulls, the testes and accessory sex glands may be affected and reveal inflammatory changes. Infected bulls may shed brucella in their semen, seminal fluid and urine, and therefore in infected herds they should always be viewed with suspicion, particularly if artificial insemination using their semen is contemplated. The risk of introducing the disease into a herd through embryo transfer is negligible. Brucella abortus is sensitive to pasteurization temperatures, and its survival outside the host is largely dependent on environmental conditions. It may survive in an aborted fetus in the shade for up to 8 months, for 2 to 3 months in wet soil, 1 to 2 months in dry soil, 3 to 4 months in faces, and for 8 months in liquid manure stored in tanks. Generally, removal of infected animals from contaminated premises for 1 month is sufficient to prevent infection, provided the facilities have been properly disinfected. A contaminated environment or equipment used for milking or artificial insemination are further sources of infection. Permanent calving camps and lush pastures, particularly if they are wet and muddy, may play a very important role in the spread of the disease. Large numbers of organisms are shed from the reproductive tract when infected cows abort. In those cows that lactate following abortion, milk, including colostrum, is an important source of infection, and bacteria may be excreted intermittently in milk throughout the lactation period. Urine and feces of infected cattle are less important sources of the bacterium. The fluid in hygromas caused by B. abortus infection may contain large numbers of organisms, but, because they are restricted to the lesion, they do not seem to be important in the spread of the disease. There is a reduction in the numbers of organisms shed in the months following calving and abortion, and cows usually eventually become non-infective until the next pregnancy, when there is again a rapid increase of Brucella organisms in the reproductive tract. During subsequent gestations, there is invasion of the uterus and allantochorion, but abortion rarely recurs. 90% of infected cows remain chronically infected. The infection may persist for life confined to the udder and target lymph nodes. Although B. abortus has been isolated from ixoid ticks and their eggs in Brazil, ticks probably do not play an important role in the transmission of the disease. Brucella abortus infection in sheep and goats may occasionally cause them to abort, but the infection does not spread in these species and they are apparently not a real danger to cattle unless there is close association between the species. Horses become infected particularly by ingestion of Brucella abortus contaminated feed. In this species the organisms localize in bursae, tendons and joints, and they are thus an unlikely source of infection for cattle. In humans, Brucellosis or undulant fever caused, among other Brucella spp, by B. Abortus is chiefly an occupational disease, occurring most often in veterinarians, stock inspectors, abattoir workers, laboratory personnel and farmers who become infected by contamination of abraded or intact skin or mucous membranes, or by inhalation during contact with infected cattle, fetuses or fetal membranes, and calves. Humans may also become infected after ingesting unpasteurized dairy products containing field strains of B. abortus or after inadvertent self-inoculation with B. abortus strain 19 vaccine. Pathogenesis. The establishment of infection is influenced by the size of the infective dose, virulence of the bacteria, and the resistance, age, sex and reproductive status of the animal. Brucella abortus readily penetrates mucous membranes such as those of the pharynx and alimentary tract. Survives and multiplies particularly in cells of the reticuloendothelial system. After penetration, the organisms are phagocytosed by neutrophils and macrophages that carry them to the regional lymph nodes, where they multiply and induce lymphadenitis that may persist for months. Multiplication of the organisms here may be followed by bacteremia, which may persist for several months, resolve itself, or be recurrent for at least 2 years in 5 to 10% of animals. Recurrence occurs particularly during pregnancy. During the bacteremic phase, organisms are carried intracellularly in neutrophils and macrophages or free in the plasma and localize in various organs, especially the uterus, udder and mammary lymph nodes. Localization can occur in other lymph nodes and the spleen, and in bulls in the testes and male accessory sex glands. Occasionally bacterial localization occurs in synovial structures, causing purulent tenovaginitis, arthritis or bursitis. Localization of the infection in the endometrium of the uterus and in fetal membranes of cattle appears to be the result of the special affinity of the organism for erythritol, elevated levels of which occur in the placenta and fetal fluids from about the 5th month of gestation. The chorionic epithelium becomes infected and it extends to the placental stroma, blood vessels and ultimately, to the fetus. There is considerable variation in the uterine and placental lesions in both natural and experimental B. abortus infections, and fetuses that become infected late in gestation may be aborted without any grossly recognizable placental lesions. Depending on the severity of the placentitis, abortion, premature birth or the birth of a viable or non-viable calf may result. Erythritol is a Brucella preferential carbon source. The placenta and the male genital organs and fluids also have high concentrations of glycerol, lactate, and glutamate. It seems therefore plausible that there is a correlation between the presence and abundance of privileged nutrients in the male and female genital organs and the nutritional preferences of Brucella in some simple defined media. The abundance of erythritol in the pregnant uterus results in the massive multiplication of Brucella organisms in this organ. The growth of most vaccine strain 19b. abortus organisms, however, is generally inhibited by the presence of erythritol, but tolerance to erythritol by some strain 19 variants may be the cause of occasional persistent infections and abortions. During animal gestation, Brucella will replicate in the placental trophoblast during middle and late gestation after the cells have actively begun secreting steroids. The mechanism leading to abortion after mid-gestation in brucellosis is not known. Infected trophoblasts produce cortisol, a steroid hormone not produced in the non-infected placenta. This production of cortisol, coupled with increased levels of estrogen and prostaglandin synthesis and decreased production of progesterone, mimics the hormonal changes occurring at term in non-Brucella infected cattle and leads to the initiation of parturition. Up to 35% of cows may be resistant to infection with B. abortus because their macrophages have a greater ability to kill B. abortus than that possessed by susceptible cows. The level of macrophage function, which is reduced in susceptible cows, plays a role in the establishment of chronic infections. This enhanced macrophage killing activity is significantly greater in cows that are genetically resistant to infection, including that caused by Mycobacterium bovis, S. dublin and S. typhimurium as well as B. abortus. The bovine RAMP1 gene, the homologue of the murine tuberculosis resistance gene, has been identified as a major candidate for controlling the in vivo resistant phenotype to Brucella infection. It has been demonstrated in a murine macrophage cell line transfected with the resistance and susceptibility associated alleles of the bovine RAMP1 gene that these alleles severely affect the control and replication of B. abortus. Phagocytes have, on the one hand, developed antimicrobial defense mechanisms, such as oxidative burst, acidification of phagosomes, or fusion of phagosomes with lysosomes, to eliminate pathogens, while on the other, facultative intracellular bacteria have developed strategies counteracting the host cell defenses, resulting in intramacrophage survival. Recent studies have revealed that caveolae or lipid rafts anchored in the membrane of macrophages are implicated in the entry of Brucella into murine macrophages and mediate an endocytic pathway that avoids fusion with lysosomes. It has been shown that in human macrophages phagosomes rapidly acidify to a pH of 4,0 to 4,5 following Brucellosis infection, and that this early acidification is crucial for intracellular replication, as neutralization results in bacterial elimination. In addition, if the phagosome membrane is disrupted, then Brucellosis fails to multiply intracellularly. These results highlight that an intact, acidic phagosome called the brucellosome is important for the intracellular trafficking of the Brucella-containing vacuole. A series of genes are involved in the adaptation of Brucella to three major stress conditions within the phagosome: acid stress, starvation and low oxygen tension. The Brucella intracellular pathogenesis, with emphasis on bacterial exploitation of the host endoplasmic reticulum-associated functions, and how autophagy-related processes contribute to the bacterium's intracellular cycle, has been recently reviewed. Cytokines such as IFN-gamma, TNF-alpha, IL-2 and IL-12 control the intracellular growth of Brucella. Among these cytokines, the most important is IFN-gamma, which strongly activates macrophages and induces enhanced intracellular killing of Brucella. In contrast to what is observed in murine macrophages, Brucellosis does not induce the production of TNF-alpha in human macrophages. Interleukin-2 favors Brucella infection by impairing a protective host immune response. In non-phagocytic cells, such as HeLa epithelial cells, the Brucella bacterium initially interacts with compartments of the early endocytic cascade, then rapidly segregates from this intracellular pathway and associates with the autophagocytic cascade. During the late stages of infection, Brucella will proliferate within the endoplasmic reticulum of host cells. They replicate extensively intracellularly, without inducing obvious damage to the infected cell, and therefore seem to promote the survival of the cells for their own benefit. By studying Brucella infection in pregnant mice, it was discovered that Brucella abortus kills trophoblasts, which are important cells for maintaining pregnancy. This killing required an injected bacterial protein that triggered an endoplasmic reticulum stress response in the trophoblast. In the bovine uterus, this extensive replication does lead to cell necrosis and acute inflammation, and to the release of huge numbers of bacterial cells from both the trophoblasts and fetal tissues. Clinical signs. The length of the incubation period of bovine brucellosis varies considerably. The incubation period has been variously defined as the period between exposure and abortion. In bulls, this period is even more imprecise, as serological evidence of infection may be equivocal or lacking, and clinical signs may be absent. The length of the incubation period is also affected by the size of the infective dose, and the age, sex, stage of gestation, and immunity of the infected animal. In cows that do eventually abort, the usual length of the incubation period varies according to the time at which infection occurred. Cows infected at service abort after an average interval of 225 days, whereas those infected at 7 months gestation abort about 50 days later. Congenitally infected calves may remain seronegative for at least 18 months, after which they may manifest clinical signs. The longest recorded incubation period in a cow is 9 years. The abortion rate in infected herds is dependent on many factors and varies according to the susceptibility of the pregnant animals, management practices, the severity of the challenge, the period for which the herd has been infected, and various environmental factors such as the quality of pastures, which may affect cattle density, the climate and the topography. In fully susceptible herds, abortion rates vary from 30 to 70%. Caution should be taken when interpreting reported proportions of cows that abort following herd infection, as this proportion should ideally be calculated as the ratio of cows that abort to cows that are pregnant and not to the total number of cows in the herd. The actual proportion of cows that are pregnant when the herd became infected is highly variable and often unknown. As a result, the ratio of abortions to total herd size, rather than to number of pregnant cows, is often reported. Increased public awareness, veterinary intervention, improved management practices and vaccination have all contributed to making the disease in these herds assume a more insidious, chronic form. In such herds, which are often closed, very few or no abortions occur and the disease is almost impossible to recognize clinically. Abortions typically occur at approximately 5 to 7 months gestation, although some occur earlier or later. Weak, full-term calves that often die shortly after birth are sometimes encountered. About 20% of infected animals do not abort, while 80% of animals that abort as a result of B. abortus infection do so only once. The placenta is not consistently retained after abortion, but when it is retained, metritis is common. Early abortion may result in a considerable reduction in the milk yield. Infection of the udder is clinically inapparent and the organ appears to be normal when palpated. In bulls, acute to chronic, unilateral or bilateral orchitis, epididymitis, and seminal vesiculitis occasionally occur. The scrotal circumference may be normal or severely increased. Strain 19 vaccine may also cause orchitis. Uni or bilateral hygromas, especially of the carpal joints, may be evident in some animals in chronically infected herds, or may occasionally follow inoculation of heifers with strain 19 vaccine. Progressive, erosive, non-suppurative arthritis of the stifle joints has been reported in young cattle from brucellosis-free herds that were vaccinated with strain 19 vaccine. Lesions. Regardless of the route of infection, the organism provokes regional lymphadenitis characterized by reticuloendothelial cell and lymphoid hyperplasia, as well as the infiltration of large numbers of mononuclear cells and some neutrophils, and a few eosinophils and plasma cells. Other lymph nodes in the body and the spleen may be affected later in the course of the infection, but to a lesser degree. There is considerable variation in the severity of the uterine lesions at abortion. As the disease progresses, lesions advance from acute, mild to severe, to chronic endometritis. Microscopically, the endometrium is infiltrated by lymphocytes and plasma cells, and some neutrophils. Microgranulomas may be scattered in the endometrium. The most significant lesions after experimental infection at 6 to 7 months of gestation were necrotizing and suppurative placentitis and lymphhistiocytic mastitis in cows, and fibrinous pleuritis, fibrinous pericarditis and bronchopneumonia in aborted fetuses, premature expulsion of a non-viable fetus more than 15 days before the predicted date of delivery. The chorion is not uniformly affected and large parts may appear quite normal. The lesions in and at the periphery of the cotyledons, as well as those in the intercotyledonary area, vary in extent, appearing to be most severe adjacent to cotyledons. The affected cotyledons, or parts of them, are covered by a sticky, odorless, brownish exudate, and are yellowish-gray as a result of necrosis. Parts of the intercotyledonary placenta are thickened, edematous, yellowish-gray and may contain exudate on the surface. Microscopically, the stroma of the chorion is infiltrated by numerous mononuclear cells and some neutrophils. Some chorionic villi are necrotic, while a fibrinopurulent exudate and desquamated necrotic chorionic epithelial cells are accumulated between the villi. Many of the chorionic epithelial cells are packed with numerous intracytoplasmic bacteria. Vasculitis, sometimes accompanied by thrombosis, may be evident in the chorion. Some aborted fetuses have varying degrees of subcutaneous edema and blood-tinged fluid in the thoracic and abdominal cavities, while the abomasum content is sometimes turbid, bright yellow and flaky. In some fetuses, grayish-white foci of pneumonia of 1 mm or larger in diameter may be present, particularly in the apical lobes. Fibrinous pleuritis sometimes accompanies the pneumonia. The liver is usually enlarged, discolored orange-brown, and its surface may have a slightly uneven appearance. Many fetuses show no gross changes. Microscopically, most aborted fetuses reveal multifocal bronchopneumonia, characterized by the accumulation of cellular debris, neutrophils and macrophages in the lumen of the bronchi and bronchiole, patchy desquamation of bronchial epithelial cells, and mild to moderate infiltration of mononuclear cells and some neutrophils in the alveolar septa. Vasculitis of some of the pulmonary vessels may be seen. Isolated small foci of necrosis or microgranulomas are often found in the liver, but may also occur in the lymph nodes, spleen and kidneys. In most aborted fetuses it is not possible, or very difficult, to demonstrate organisms in tissue sections, notwithstanding that they may have been specially stained for Brucella. However, it is easy to demonstrate the organisms in smears made from the abomasum content or wall that have been stained with stamp stain. The udder in infected ruminants does not show any gross lesions, although the supramammary lymph nodes may be enlarged. Microscopically, infection of the udder is characterized by lymphoplasmacytic and histiocytic interstitial mastitis, while the regional lymph nodes show lymphoid hyperplasia, medullary plasmacytosis and sinus histiocytosis. Acute orchitis is characterized by multifocal or diffuse necrosis of the testicular parenchyma, and focal, necrotizing epididymitis may occur. Microscopically, the seminal epithelial cells are necrotic and desquamate, large numbers of organisms are present in them, while numerous leukocytes, particularly neutrophils, and fibrin occur in the affected tubules and interstitial tissues. In the chronic stage, spermatic granulomas develop in the testicular parenchyma and epididymis in response to dead sperm. In horses, infection may be present in the bursa between the nuchal ligament, the atlas and axis, causing pole evil. Fistulous withers caused by B. Abortus is characterized by inflammation of the bursa between the nuchal ligament and the dorsal spines of the thoracic vertebrae. Chronic draining sinuses are formed in both conditions. Brucella in cattle may localize in the carpal and other bursae and induce the formation of hygromas in which large numbers of the pathogen may be found. Diagnosis. In order to develop sensitive and specific diagnostic tests and more efficacious vaccines and to explore new therapeutic approaches in both human and animal brucellosis, it is important to assess both innate and acquired specific immune responses directed against Brucella. Because Brucella controls the traffic of its intracellular vacuole in phagocytes and in the trophoblast of the pregnant uterus. Antibodies, while contributing to the protection against Brucella if present when infection occurs, for example after vaccination, are not as effective as that provided by cell-mediated immunity. The humoral immune response is directed against the OPS moiety of the smooth lipopolysaccharide, SLPs. The Brucella anti-protein antibody response is often delayed when compared to the anti-SLPs response and is limited to animals that develop an active Brucella infection. Is evidence for a protective role of humoral antibodies directed either against LPs, or outer membrane proteins. The antibody response has been extensively used for the serological diagnosis of bovine brucellosis. Cellular immune responses contribute to the control of virulent and attenuated, vaccine strains, Brucella, more than the humoral response does. It has been shown for many intracellular bacteria and protozoa that IFN gamma activates the microbial killing activity of macrophages. Unfortunately, only few specific bacterial antigens involved in stimulating protective cellular immunity against Brucella are known. Murine macrophages infected with Brucella produce cytokines such as TNF-alpha and IL-12. IL-12 seems to be critical in the mouse model because its depletion induces an exacerbation of the infection as well as an inhibition of the IFN gamma production. IFN gamma is an important component of the type 1 cellular responses referred to nowadays as Th1 cell responses. These responses are principally characterized by the production of T1 cytokines, and IFN gamma and IL-2, by CD4 plus T helper cells and CD8 plus T cytotoxic cells as well as Ig2A secretion by plasma cells. For extracellular bacteria or toxins, a Th2 cell response will be induced. This response is principally characterized by the production of Th2 cytokines, i.e., IL-4, IL-5 and IL-10, by CD4 plus T helper cells as well as Ig1 and Ig-A secretion by plasma cells. According to the triggering mechanism, a precursor T helper cell will differentiate into either a Th1 or a Th2 cell. Live brucellosis vaccines are more effective than inactivated vaccines because the latter tend to induce a Th2 response. Therefore, it is important to identify bacterial proteins that induce a Th1-mediated response, and it is critical to determine how the host first recognizes and then eliminates Brucellae. However, although inducing a cellular immune response, few of these individual proteins induce host protection. Other T lymphocyte subsets, such as gamma-delta T lymphocytes, may be important as mediator, secretion of IFN gamma and IL-2, or lytic effector cells in innate and acquired immunity. Bovine gamma delta T cells from cattle infected with Mycobacterium bovis or vaccinated with a killed Leptospira petersonii. Adjuvanted vaccine, recognize specific antigens and produce IFN gamma as part of an acquired immune response, which could play a role in protection. Since of the variable incubation period and the often subclinical nature of the disease in most animals, a definitive diagnosis should be based on the isolation and identification of Brucella abortus and on positive serological results based on the detection of antibodies in blood, milk, whey, vaginal mucus, or seminal plasma. The isolation of Brucella abortus is important when dealing with specific groups or herds of animals when there is doubt about their infection status. When a herd is known to be infected with B. abortus and where its environment is infected, serological reactors are viewed in a different light from those in a herd in which reactions might be due to the use of strain 19 vaccine. It is preferable to interpret serological test results according to the known infection status of the herd. Nevertheless, failure to isolate Brucella in a herd may result in difficulty in interpreting serological results. Diagnostic tests for bovine brucellosis are subdivided into 3 groups. Tests for the demonstration of B. abortus organisms. Tests which detect immunoglobulins. Tests which detect allergic reactions to B. abortus. The ideal diagnostic test should detect infection early, during the long and variable incubation period, not be influenced by the presence of nonspecific antibodies, detect carriers, and differentiate between responses to vaccination and those due to field infection. There is no single available test that completely covers all these requirements. The tests described below are standard tests that are applied worldwide. Minimal requirements to ensure quality control of the reagents for the demonstration of B. abortus and the methods employed for the serological and allergic diagnosis of bovine brucellosis can be found in standard laboratory textbooks. Microscopic examination. Using Stamp's modification of the Ziehl-Neelsen stain, B. abortus stains red, against a blue background in tissue sections and smears. However, this color reaction is not specific for Brucella spp, as Coxiella burnettii, Chlamydophila abortus and Nocardia spp. Are also weakly acid-fast. Nocardia spp. Can be differentiated from these organisms on morphological grounds, but it is difficult to differentiate Chlamydophila abortus and Coxiella burnettii from Brucella spp. Beyond any doubt. Culture and typing. The specimens of choice include fetal membranes, lungs, stomach content, liver and spleen of aborted fetuses and full-term calves, and from live cows' uterine discharge, milk or colostrum. The supramammary lymph node is the most suitable specimen from carcasses of adult animals, but the retropharyngeal, mandibular, iliac, or the prescapular and parotid lymph nodes may also be collected. Other specimens that may be submitted include uterus, milk, udder tissue, fluid aspirated from hygromas, male accessory sex glands and testes. Isolation may also be attempted from the semen or seminal plasma of infected bulls. The supramammary lymph nodes are preferred for isolation of B. abortus from animals that have been slaughtered, and a 90% recovery rate from infected animals may be achieved. Recovery of B. abortus on culture from infected adult cows may approach 100% using the supramammary, parotid, mandibular and subiliac nodes. Culture and typing. Isolation of the pathogen from infected heifers is best achieved by the culture of the mandibular lymph nodes, and this probably reflects the primary route of infection through the conjunctiva or the nasal or oral mucosa. When specimens of large groups of cows, 5 or more, must be cultured, collection of supramammary lymph nodes should be sufficient. In order to get valuable epidemiological information, isolated brucellae have to be typed, species and biovar, according to the standard biotyping methods described in WA textbooks. Besides this method, molecular typing methods such as MLVA, multiple locus variable number tandem repeat analysis, and multi-locus sequence typing, MLST, as well as whole genome sequencing, WGS, are nowadays used and have provided important information related to Brucella phylogeny, taxonomy and epidemiology. Representative Brucella abortus strains isolated worldwide could be subdivided by MLVA into 3 clades congruent with the clades B, C1, and C2 of multi-locus sequence typing. Polymerase chain reaction. Numerous polymerase chain reaction, PCR-based assays have been developed for the identification of members of the genus Brucella. Certain circumstances require the identification of the Brucella spp involved. For epidemiological traceback, strain-specific identification is helpful. Several strategies have been explored to differentiate between Brucella spp and biovars, including locus-specific multiplexing, PCR-RFLP at the OMP2 locus 44 and AMOS PCR based on IS-711 insertion sequence, allowing the differentiation of the vaccine strains 19 and RB51. Real-time PCR was developed in a multiplex format in 2004, allowing the rapid identification of Brucella spp, Brucella abortus, and melitensis in a single test. Multiplex PCR assay, Brucellosis, for rapid and simple one-step identification of Brucella has been developed and validated in 2008. It can identify and differentiate in a single step most Brucella spp, as well as the vaccine B. abortus strains S19 and RB51 and the vaccine B. melitensis strain Rev.1. Tests for the detection of specific immunoglobulins. The serological diagnosis of brucellosis began in 1897 with the development of an agglutination test by Wright. Problems such as false positive serological reactions resulting from, for example, exposure to cross-reacting microorganisms, were encountered. At present, several serological tests are used worldwide. The outer membrane of smooth Brucella spp strains is composed of phospholipids, proteins and SLPs. Most of the serological tests, particularly those using whole cell suspensions as antigen, such as the slow, TUBE, agglutination test, SAT, the Rose-Bengal test, RBT, the complement fixation test, CFT, most enzyme-linked immunosorbent assays, ELISA, and the milk ring test, MRT, have been developed to detect antibodies directed against the OPS moiety of the SLPs. These tests are suitable for use in surveys, large-scale campaigns and in control/eradication programs of the disease as well as for trading purposes. Inexpensive, rapid and simple screening tests with high sensitivity followed by more specific confirmatory tests in case of positivity in the screening test, are used in control programs. Tests for the detection of specific immunoglobulins. The ability of individual tests to correctly identify infected or non-infected animals, i.e., their sensitivity or specificity, can be enhanced by combining two or more tests and interpreting their results in parallel or in series, depending on whether the objective is to increase the negative or the positive predictive value. An epidemiological inquiry and a risk factor analysis also need to be undertaken. In acute bovine brucellosis, an increase in the level of IgM in the serum is the first evidence of antibody response. IgG soon becomes the predominant antibody and usually persists for as long as the animal remains infected while the IgM levels wane. Of the two subclasses of IgG in the serum, IgG1 is the most abundant and predominant agglutinating and complement-fixing antibody. Rose-Bengal test. The RBT is a modification of the plate agglutination test. The antigen, which has been stained with Rose-Bengal stain, is buffered at a pH of 3,65. At this level of activity, non-specific agglutinins are destroyed and IgG, normally the most abundant antibody in the serum of infected animals, agglutinates strongly. The test is inexpensive and easy to perform. False negative results are rare and are usually obtained during the more chronic stages of the disease. Despite improved specificity at an acid pH, a high percentage of false positive reactions occur usually due to the presence of IgM as a result of strain 19 vaccination. This test is prescribed for international trade in cattle by the WA. Complement fixation test. This test is regarded throughout the world as being the confirmatory test for the serological detection of infected animals. It has been modified, standardized and adapted to a microtiter system. Unlike the SAT, the titers do not wane as the disease becomes chronic. Results are expressed in international units, IU, and a cutoff point of 20 IU has been defined, which is rigorously applied where strain 19 vaccine has not been used for several years, as is the case in most of the European Union, EU, the USA and Australia. Its strict application in countries enforcing compulsory strain 19 vaccination may often be problematic and sometimes leads to unacceptably large numbers of false positives, because vaccination induces serological titers. As a consequence, considerable expertise and experience are needed to certify herds or individual animals free of brucellosis when they are classified as positive by the test. As a rule, vaccine titers tend to decline faster than those due to infection with wild strains. The decline in titers is also dependent on the vaccine dose. Although the CFT is useful when differentiating CAFOD vaccination titers from those due to wild strain infections, there may be difficulty in distinguishing vaccine reactions from those caused by wild strains when animals have repeatedly been vaccinated, although CAFOD vaccination alone is considered to be adequate, or after they have become sexually mature. This test is prescribed for international trade in cattle. Slow, TUBE, agglutination test, SAT in a number of countries. The SAT was and still is used as a screening test for eradication purposes. It is considered to lack specificity and some authors discourage its use, at least for trading purposes. Nonspecific agglutination in Syrah is decreased by the addition of EDTA with no reduction in B. Abortus agglutination titers of Syrah from infected cattle. The SAT EDTA is a very specific test and particularly useful in detecting new infections as early as 2 weeks after infection, as demonstrated in experimental conditions, but its usefulness in herds that are chronically infected is more limited because some infected animals will be classified as negative by this test because the infection is in a chronic phase. Indirect enzyme-linked immunoabsorbent assay. Indirect enzyme-linked immunosorbent assay, IELISA, is more sensitive for detecting antibodies to Brucella spp than are the RBT, SAT and CFT, but great care must be exercised in animals vaccinated with strain 19 vaccine. It has been suggested that this test could replace not only the currently used confirmatory CFT, but also the two in-use screening tests, namely the RBT and SAT. The IELISA has been used successfully throughout Europe in strategies aimed to substantiate and to maintain the status of brucellosis-free countries. When the herd prevalence is low, less than 1%, a pooled Syrah approach could be adopted by policymakers to reduce the cost of the surveillance program and increase the number of samples being tested. IELISA can also be used on individual or bulk milk samples. This test is prescribed for international trade in cattle by the WA. Competitive enzyme-linked immunoabsorbent assay. The basis of this assay is the use of a selected monoclonal antibody, mAb, that competes with low-affinity antibody. The competitive enzyme-linked immunoabsorbent assay, SELISA, using a mAb specific for one of the epitopes of the Brucella abortus OPS has been shown to have higher specificity than the IELISA. The SELISA was reported to be able to eliminate cross-reaction problems in serological tests induced by strain 19 vaccination or infection with cross-reactive bacteria. Unfortunately, the SELISA only partially solves the problem. Indeed, persistent competing antibodies have been observed after Yersinia enterocolitica O, infections, and vaccination with strain. However, residual antibody activity due to vaccination or cross-reactive infection was less persistent than with the other tests. In a recent study, none of the SELISA kits studied could be recommended as a single screening test because of their low specificity. This assay is prescribed as an alternative test for international trade in cattle by the WA. Fluorescence polarization assay. The fluorescent polarization assay, FPA, is a simple and rapid technique for measuring antigen/antibody interaction and may be performed in a laboratory setting or in the field. The mechanism of the assay is based on random rotation of molecules in solution. A fluorochrome labeled antigen of small molecular weight, a fragment of the O polysaccharide, OPS, of B. Abortus SLPs, for example, is added to serum or other fluids to be tested. If antibody is present, attachment to the labeled antigen will cause its rotational rate to decrease and this decrease can be measured. The FPA has received very promising reports. This test is prescribed as an alternative test for international trade in cattle by the WA. The milk ring test is used to detect antibodies in milk. The development of a positive reaction is dependent on two reactions. Fat globules in the milk are aggregated by milk antibodies, fat globule agglutinins, and stained Brucella cells, antigen, which are added to the milk, are agglutinated by the Brucella antibody, fat globule complexes, which rise to form a colored cream layer at the top. Despite its reduced sensitivity in large herds, the MRT has been very successfully used to monitor the brucellosis-free status of dairy herds. Several countries have replaced the MRT by a milk ELISA. Although this latter technique has not been standardized, it is prescribed for international trade in cattle by the WA. Allergic reaction to Brucella abortus. A skin test for the diagnosis of brucellosis has been used in extensively managed herds in New Zealand. The sensitivity of the test was considered to be low at the animal level and its specificity exceeded 99%, and thus it was claimed that this test is a useful low-cost method of identifying infected herds rather than individual animals. This test has recently been re-evaluated in the EU because of the emergence of false positive serological reactions, FPSR, i.e., nonspecific reactions in all serological tests, that have emerged throughout Europe. These have been documented in Belgium and France since 1990, affecting up to 15% of the herds tested in some regions that are free of brucellosis. Yersinia enterocolitica infections have been shown to be responsible for these FPSR. It is now a recommended herd test for brucellosis by the WA and as an official test in the EU, Directive 64/432, particularly when monitoring is made difficult due to nonspecific brucellosis serological reactions. However, CAFOD vaccination using strain 19th of may complicate interpretation of the Brucella test by inducing prolonged sensitivity to Brucella allergens. A diagnosis of bovine brucellosis based on in vitro antigen-specific IFN gamma production, which can be regarded as an in vitro correlate of the brucellosis skin test, has been developed. Unfortunately, this test has been shown to be less specific than the brucellosis skin test. Differential diagnosis. Numerous infectious agents may cause fetal loss and abortion in cattle. A multidisciplinary approach in terms of diagnostic tests is necessary to make a definitive diagnosis. Microscopic examination of smears or histopathological sections, particularly of the placenta, stained by stamp method, may present difficulties in differentiating Brucella abortus morphologically from Coxiella burnettii or Chlamydophila abortus. Brucella abortus can be distinguished by PCR, fluorescent antibody techniques and serological tests. Apart from these agents, there are numerous viral, bacterial and parasitic causes of abortion in cattle that should be considered. Brucella abortus may cross-react serologically with Escherichia coli serogroup 0, Y, Enterocolitica cerevaro, Salmonella serotypes of the Kaufmann white group N, Francisella tularensis, Pseudomonas multifila, and Vibrio cholera 49 because the immunodominant O chain of SLPs of these bacteria contains antigenic motifs, epitopes, that may be detected in serological tests for brucellosis using whole B. abortus cells or SLPs extracts. Treatment. Cattle suffering from bovine brucellosis are not treated. Vaccination. In 1906, Bang observed that cattle could be protected against brucellosis by vaccinating them with live cultures. 3 strains of Brucella abortus have been used for the preparation of vaccines and have been studied extensively. Strain 19, a smooth live attenuated vaccine. Strain 4520, as a rough-killed vaccine. Strain RB51, as a rough live attenuated vaccine. The minimal requirements for vaccine production have to be followed and each batch of live vaccine must conform to the minimum standards set by the WA. These include viability, pathogenicity and ability to immunize guinea pigs and or mice against challenge with a virulent strain of B. abortus. The planning and management of control or eradication programs for brucellosis have been investigated in many countries, and by the World Health Organization. In the EU, a task force for monitoring disease eradication in the member states was created in 2000 with the objective, as far as bovine brucellosis is concerned, of expanding eradication schemes and improving the cost-benefit ratio of eradication programs co-financed by the EU. Strategies based on the prevention of the spread of the disease between animals, the monitoring of brucellosis-free herds and zones, the elimination of infected animals by test and slaughter, strict one-way movement of infected and suspect cattle, mass immunization to reduce the infection rate, and supporting specific education and training programs, have all received attention. It is not appropriate to be prescriptive when recommending a strategy for the control or eradication of brucellosis that will be applicable or practical in all circumstances. Broadly based national strategies and policies will invariably differ in time according to the situation in each instance. The approaches to be adopted when attempting to eradicate the disease will vary markedly according to whether one is dealing with an infected herd or an uninfected, vaccine reactions, herd. Control measures on individual farms are also frequently affected by a variety of other factors. These include the infection rate in the herd, duration of infection, the abortion rate, whether an extensive or intensive farming system is being practiced, rainfall, whether it is a beef or dairy herd, vaccine cover, the availability of, or necessity for, replacement stock, the financial situation of the producer, his proximity to a veterinary laboratory, neighborhood cooperation, milk and slaughter quotas, slaughter prices of dairy cows, the availability of camps and suitable calving facilities, availability of veterinary guidance, and the handling facilities necessary to collect blood samples from large groups of cattle. The crucial principles to the successful control or eradication of brucellosis. Reliable veterinary services. Reliable diagnostic techniques. Close cooperation and joint effort by the owner, laboratory, veterinary services and veterinarian. Good records. Education of the farming community and public awareness. Established procedures for handling the disease. Controlled movement of cattle, and permanent marking of infected and vaccinated animals. Vaccination in conjunction with other measures can be used effectively in the control or eradication of brucellosis and, in general, it is recommended that all heifer calves be immunized where the prevalence of brucellosis in an area, region, province or country is high, and that cessation of immunization with strain 19 vaccine only be considered when the prevalence of the infection is reduced to 0.2% or less. At that time, a test and slaughter, management and hygiene programs should be implemented. Control. Brucella abortus remains endemic in low-resource countries in which an intensive vaccination program that reaches optimal coverage may not be realistic in the short term. In fact, it has been suggested that in some of these countries the prevalence of infection may be increasing as more intensive livestock systems emerge. Given the potential economic and public health impact of infection, it is important that progressive control strategies, combining livestock vaccination, gradual culling and environmental sanitation, that can be sustained with limited resources are considered. The reduction in numbers and the rapid elimination of all infected animals are essential in the control or eradication of brucellosis. All calves in infected herds should be regarded as potential sources of infection. The 2-year breakdown syndrome, that is those previously infected herds which have apparently been clear for about 2 years and in which a breakdown occurs after this period, has been ascribed to latently infected calves that remain serologically negative until at least mid-gestation or later during their first pregnancy. Cows should calve in an environment that can be thoroughly cleaned and disinfected with 2.5% formalin. Wet and well-grassed calving camps should be avoided, and vehicles used for transporting infected animals should be disinfected after use. Isolation of cows prior to and after parturition contributes significantly to the control of brucellosis, and, in addition, cows that abort should be isolated immediately and tested serologically soon after they aborted. If negative, they should be retested 2 to 3 weeks later. Animals testing positive should be culled. Fetuses, placentas and discharges must be disposed of, preferably by incineration. It is important to maintain regular blood testing in infected herds. Serological tests at intervals of 2 to 3 months and bacteriological examination of aborted fetuses, placentas, lochia and milk are often of great benefit. Milk from negative animals should be tested regularly and at short intervals, monthly, by the MRT. All bulls from infected herds should be viewed with suspicion and the diagnosis should be based on clinical examination, including palpation of the seminal vesicles and ampullae, serological and allergenic tests, and on bacteriological examination of semen. It is important to prevent, or, in outbreaks, at least limit the spread of the disease. Brucellosis in small ruminants, caprine and ovine brucellosis. Brucella melitensis infections. Brucella melitensis is the main causative agent of caprine and ovine brucellosis. It is also pathogenic for several other mammal species including humans. Infected cows may abort and shed Brucella melitensis in their milk. Brucellosis in small ruminants is characterized by one or more of the following: abortion, reduced milk yield and retained placenta, particularly in goats, in females, orchitis and epididymitis in males, and, rarely, arthritis in both sexes. It is worth noting that brucellosis caused by Brucella abortus infection in small ruminants seems to be an important problem in Africa, whereas Brucella suis infection is rare in small ruminants. The organism responsible for Malta fever in humans, discovered by Sir David Bruce on the island of Malta in 1887. It had become evident to him and his co-workers that the disease was not transmitted from person to person. Zammit, a member of the Mediterranean Fever Commission, determined in 1905 that Brucella melitensis was the cause of contagious abortion in goats on the island and that humans contracted the disease by the consumption of infected fresh goat's milk and milk products. Brucella melitensis is morphologically and tinctorial indistinguishable from B. abortus and B. suis. Species identification can be based on lysis by phages, on biochemical tests, such as oxidase, urease and catalase, or molecular tests. Unlike abortus, growth of Brucella melitensis is not dependent on an atmosphere of 5 to 10% CO2, although there might be some exceptions. The identification of a Brucella species to the biovar level is currently performed by four main tests, namely, CO2 dependence, and production of hydrogen sulfide, thionine and basic fusion, sensitivity, and agglutination with monospecific A and M anti-Syrah. The three biovars, 1, 2 and 3, recognized for Brucella melitensis show no difference in pathogenicity. Brucella melitensis biovar 3 appears to be the biovar most frequently isolated in Mediterranean countries, while biovar 1 seems to predominate in Latin America. In a multi-locus sequence analysis, MLSA, study, Brucella melitensis isolates clustered into three distinct lineages that correspond to the Americas, West Mediterranean, and East Mediterranean lineages described earlier using multiple locus variable, number of tandem repeats, analysis, MLVA, based studies. Although a few isolates from the USA and Argentina were included in the Americas clade, more than 60% of these isolates clustered with strains of African origin. African isolates fit into the West Mediterranean and East Mediterranean lineages. They may represent lineages that are endemic in Africa. In agreement with the above-mentioned MLVA study, strains falling in the West Mediterranean lineage were isolated exclusively in Italy, France, the former Yugoslavia and Germany. In contrast, the East Mediterranean lineage, while including isolates from Greece, Turkey, Cyprus, and the Balkan states, extends geographically more widely into the Middle East and Asia, Thailand, India, Pakistan, Mongolia, and Afghanistan. While most isolates in the Americas and West Mediterranean lineages corresponded to biovar 1, 81%, and biovar 3, 64%, respectively, both lineages included isolates of all three biovars. This MLSA study concluded that the Brucella melitensis biovar concept is reliant solely on the serological reaction observed with the monospecific anti-A and anti-M Syrah and is of no epidemiological significance. Smooth, S. Brucella melitensis cultures have a tendency to undergo variation during growth, especially on subculturing, and dissociate to rough forms. These changes in colonial morphology are generally associated with changes in virulence and antigenic and immunogenic properties. Such changes may also occur during the production of the live attenuated S vaccines, particularly in the case of Brucella melitensis Reverend 1 vaccine, which may have serious consequences. Brucella melitensis wild-type strains are usually resistant to penicillin and sensitive to streptomycin, whereas the Reverend 1 vaccine strain is sensitive to benzyl penicillin and resistant to streptomycin. Microbiological test and several PCR procedures are used to differentiate field B. melitensis biovar 1 strains from Reverend. Brucella melitensis infection occurs worldwide, and is prevalent in small ruminants, particularly in the Mediterranean countries, the Middle East, some regions of Asia and Latin America. Infections have also been encountered in dromedary camels, Camelus dromedarius, Bactrian camels, Camelus bactrianus, and cattle in contact with infected sheep and goats. Northern Europe, Southeast Asia, Australia, New Zealand and Canada are believed to be free of the disease. In the USA, Brucella melitensis was detected in 1999 in cattle and goats in Texas after absence since the early 1970s, but the source of infection was not determined. All breeds of goats are believed to be equally susceptible to the infection, but despite the scarcity of data, resistance is assumed to vary with breed in sheep. Maltese sheep appear to be relatively resistant, while certain fat-tailed breeds, such as Awassi, are highly susceptible. The oral route is the main route of infection through the sucking or licking of aborted fetuses and their placentas and vaginal discharges, and the ingestion of infected milk or feedstuffs. Brucella melitensis may persist in the udder for years. Inhalation of contaminated dust is also believed to be an important route of infection. Herding of goats and sheep into stables during the night creates an ideal environment for the spread of the infection. As in the case of B. abortus infection in cattle, Brucella melitensis can be transmitted from ewes to lambs. A small proportion of lambs can be infected in utero, but the majority of Brucella melitensis infections are probably acquired by the ingestion of infected colostrum or milk. Although not proven, it is likely that a self-cure mechanism, similar to that considered to occur in calves, takes effect in most of the infected lambs. However, infections in lambs greatly increase the difficulty of eradicating this disease, as Brucella melitensis may persist until adulthood without inducing a detectable serological immune response. However, orchitis and epididymitis are common sequelae of infection, and Brucella melitensis is excreted in semen in a significant proportion of infected males, with a concomitant risk of transmission. It has in fact been shown that artificial insemination may spread brucellosis among animals. Dogs kept on farms with infected flocks may become infected and develop generalized disease, and they, especially the pregnant bitches, may spread the infection. Surprisingly, the ecological range of Brucella melitensis in wildlife is more restricted than the ranges of B. abortus and B. suis. In fact, in a study conducted in heavily infected areas in Spain, it was shown that wild ruminants were not a significant brucellosis reservoir for domestic livestock. Spillover of Brucella melitensis from infected small ruminants and cattle to a small number of wildlife species such as chamois, rupicapra, and ibex, capra ibex, in the French and Italian Alps has been documented in southern Europe. The survival of Brucella melitensis in the environment is similar to that of B. abortus. Brucella melitensis is highly pathogenic for humans, causing the disease known as Malta fever, also called Mediterranean or Undulant fever, which is rated by the World Health Organization as one of the most important zoonosis. Brucellosis in humans is also caused by B. abortus and B. suis infections, the resultant clinical disease being indistinguishable from the disease caused by Brucella melitensis. Brucellosis in humans is transmitted by ingesting infected unpasteurized milk and milk products and by direct contact with infected animals, animal carcasses and aborted material, and the organisms themselves in laboratories, as well as by accidental inoculation with the Brucella melitensis Reverend 1 vaccine stain. Worldwide, millions of humans are at risk, especially those in developing countries where the infection in animals is not well controlled. Heat treatment procedures of milk, e.g., pasteurization, are not routinely applied, and poor hygienic conditions and food habits such as the consumption of raw milk favor human infection. The survival of Brucella in milk and dairy products depends on a variety of factors. However, it is generally accepted that industrial pasteurization or prolonged boiling of milk readily kill Brucella organisms. The bacteria do not persist for periods exceeding 3 months in ripened fermented cheese. Milk or cream used in the preparation of cheese should be pasteurized to ensure its safety for human consumption. It is worth noting that humans are often the first to be reported as being infected in an area into which the disease has been newly introduced. Epidemic human infections caused by Brucella melitensis occur in people frequently in contact with infected goat herds or goat manure, as reported in Argentina. Localization of Brucella melitensis in the udder of infected camels and cattle is also a major public health problem. In laboratories handling Brucella infected or potentially infected material, it is mandatory that conditions of high-level biohazard containment are used. Pathogenesis and clinical signs. The pathogenesis of the disease is similar to that of B. abortus infection in cattle. The first sign of the presence of the disease in a susceptible herd of goats or flock of sheep is usually an abortion storm during which a high proportion of the pregnant animals abort, generally late in gestation. In some cases, particularly in nanny goats, retention of placenta and fetal membranes occurs. Milk yield following abortion is poor, and the quality of the milk may be reduced. Genital discharges of infected nanny goats are usually copious and persist for several months following abortion or kidding. Genital discharges are less copious and less persistent in infected ewes. In males, localization of Brucella melitensis in the testis, epididymis and accessory sex organs is common and may result in acute orchitis and epididymitis, and subsequently in reduced fertility. Bacteria may be shed in the semen. Arthritis is occasionally observed in both sexes. During subsequent breeding seasons, the number of animals in a flock that abort declines progressively and eventually abortions may cease to occur. Such flocks, however, remain infected for years. Persistent infection of the mammary glands with constant or intermittent shedding of the organism in the milk in succeeding lactations is frequent. Lambs from infected females may be born weak or are asymptomatic. Some of them may become persistent latent carriers of the infection. In areas where brucellosis is endemic, the prevalence of infection with Brucella melitensis in some flocks may be high even in the absence of any signs of overt disease. The macroscopic lesions in nanny goats and ewes that abort following infection with Brucella melitensis include edema and grayish-white areas of necrosis of the placenta and the presence of a brownish-red exudate between the atlantochorion and the endometrium. Microscopically, small granulomatous or necrotic foci are evident in lymphoid tissues, organs such as reproductive organs and associated lymph nodes, and synovial membranes. In pregnant females, there may be extensive necrosis of the chorioallantoic membranes accompanied by vasculitis and acute endometritis with large numbers of bacteria and necrotic villi. Desquamated trophoblast cells and a few macrophages, neutrophils and plasma cells occur in the spaces between the chorionic villi and septa. Brucella melitensis can be demonstrated by microscopic examination of smears made from vaginal discharges or swabs, placenta or abomasum of the aborted fetus and stained with stamps method. However, as morphologically similar microorganisms such as Brucella ovis, Chlamydophila abortus and Coxiella burnettii can be confused with Brucella melitensis. The most reliable and the only unequivocal method for the diagnosis of animal brucellosis is the isolation and typing of the causative bacterium using appropriate culture media. Vaginal excretion of Brucella melitensis usually persists for several weeks after abortion, while the mammary gland is the main target of infection in small ruminants. Vaginal swabs and milk are therefore the samples of choice to isolate Brucella melitensis from live nanny goats and ewes. Attempts should be made to culture the organism from the cotyledons, abomasum content and lungs of the fetus and, in slaughtered animals, from the mammary glands and supramammary lymph nodes in females, the seminal vesicles and testes in males, and the parotid, mandibular and retropharyngeal lymph nodes and spleen in both sexes. Selective media should be used for isolation of Brucella melitensis because contaminants are usually present in field samples that will overgrow Brucella melitensis colonies. Farrell's selective medium, developed for the isolation of Brucella abortus from milk, is recommended for the isolation of Brucella melitensis. However, this medium contains nalidixic acid and bacitracin which, at the concentrations used, have inhibitory effects on some Brucella melitensis strains, so it should be used with the less selective Thayer-Martin modified medium. The CETA medium, which inhibits most contaminant microorganisms but allows simultaneously the growth of all Brucella species, is the selective medium of choice for the isolation of all Brucella spp. The best diagnostic sensitivity is obtained using both the Farrell's and CETA simultaneously. Numerous polymerase chain reaction, PCR-based assays have been developed for the identification of members of the genus Brucella. For epidemiological traceback, strain-specific identification is required but, despite the considerable progress made, no technique is available for the differentiation of the three Brucella melitensis biovars. Initially all these molecular assays were performed on purified DNA obtained from previously cultured organisms. For the identification of Brucellae from field samples and food products such as milk and cheese, the removal of polymerase inhibitors is required before the implementation of PCR techniques. Real-time PCRs have been developed for the specific detection of Brucella melitensis in milk. However, these techniques need to be further validated for routine diagnostic purposes. The differentiation of the Brucella melitensis Rev. 1 vaccine strain from wild Brucella melitensis is accomplished by microbiological laboratory tests such as sensitivity to penicillin and streptomycin, size of colony, and growth on media containing dyes. Chromosomally acquired streptomycin resistance is frequently due to mutations in the RPSL gene encoding the ribosomal protein S12, and a PCR-RFLP based on a mutation in the RPSL gene has been described for the identification of the Brucella melitensis Rev. 1 vaccine strain. A multiplex PCR assay was developed and can differentiate in a single step all of the classical Brucella SPP, including those found in marine mammals and the S19, RB51, and Rev.1 vaccine strains. Several serological tests are used to diagnose brucellosis in small ruminants. The outer membrane of smooth Brucella SPP strains is composed of phospholipids, proteins and lipopolysaccharide. The epitopes involved have been located on the O polysaccharide, OPS, moiety of the SLPs, which represents the most exposed antigenic structure on the surface of smooth Brucella SPP. Most of the serological tests, particularly those using whole cell suspensions as antigen, such as the Rose-Bengal test, RBT, the complement fixation test, CFT, and most enzyme-linked immunosorbent assays, ELISA, have been developed to detect antibodies directed against OPS antigens. The RBT and the CFT are the most widely used tests for the serological diagnosis of ovine and caprine brucellosis. These tests are currently the official standardized tests used in the European Union member states, and are the tests prescribed by the WA for international trade. One of the controversial issues concerning the serological diagnosis of Brucella melitensis infection in small ruminants is related to which Brucella SPP and biovars are used in the production of the antigens. The antigenic suspensions, whole cells, used in both the RBT and CFT are made with B. abortus biovar 1. The CFT has many drawbacks such as complexity, variability of reagents, prozones, anti-complementary activity of Syrah, difficulty to perform with hemolyzed Syrah and subjectivity of the interpretation. Therefore, while the sensitivity of the RBT is sufficient for the surveillance of areas free of the disease at the flock level, the RBT and CFT should be used together in infected flocks to obtain an accurate individual sensitivity level in test and slaughter programs. Moreover, another important drawback of both the RBT and CFT is their low specificity when testing Syrah from sheep and goats that have been recently vaccinated subcutaneously with the Brucella melitensis Rev. 1 vaccine. However, when this vaccine is applied conjunctival, the interference problem is significantly reduced in all serological tests. Despite the above drawbacks, the combination of the RBT as screening test and the CFT as a complementary test has been used successfully in many countries to eradicate Brucella melitensis infection in small ruminants. The serum agglutination test is not considered to be sufficiently reliable for use in the diagnosis of infection in small ruminants. Indirect ELISAs are reported to be adequately sensitive for diagnosis. The specificity of the indirect ELISA is quite low when testing Syrah from animals that have been vaccinated recently with the Rev. 1 vaccine. Competitive ELISA and fluorescence polarization assay have been developed but not properly standardized and validated for wide use in small ruminants, and do not provide any advantages over the above-mentioned tests. Milk ring test does not give as clear-cut a reaction as it does in cattle infected with B. abortus, and it is thus not recommended as a flock or herd test in small ruminants. A technique similar to the indirect serum ELISA has also been proposed for testing individual or pooled milk samples for the diagnosis of ovine and caprine brucellosis. Allergic brucellosis skin test for the diagnosis of Brucella melitensis infection has been used with variable degrees of success to detect specific delayed type hypersensitivity, DTH, responses in sheep and goats. The only skin test allergen available, known as Brucellar gene OCB, Brucellin inro or Brucellin, is a hypertonic cytosoluble extract obtained from the rough Brucella melitensis B115 strain, and is commercially available. The site and route of inoculation of the allergen do not affect the sensitivity of the test. The method considered to be the most efficient and practical is to inoculate the allergen into the lower eyelid. Readings are made after 48 to 72 hours. The Brucellosis skin test is considered to be 100% specific when testing unvaccinated Brucella-free animals, but this is not the case when testing animals vaccinated recently with the Brucella melitensis Reverend 1 vaccine, which can remain positive in this test for as long as 2 years or more after vaccination. Control. Vaccination of sheep and goats, with the live attenuated Brucella melitensis Reverend 1 vaccine, is the only practical and effective method for reducing the incidence of brucellosis. Due to its ability to induce abortion, and the possibility of the organism being excreted in milk, it was recommended that the Brucella melitensis Reverend 1 vaccine be administered subcutaneously, in young replacement sheep and goats, prior to the first gestation, at 3 to 7 months of age. However, when Brucella melitensis strain Reverend 1 vaccine is administered by the standard method, it may induce a long-lasting serological response, in classical brucellosis tests, RBT and CFT, that could interfere in the serological diagnosis of the disease itself. In contrast, when this vaccine is administered by the conjunctival route, the immunity conferred is similar to that induced by the standard method, but the serological response evoked is significantly reduced, making it the method of choice when combining vaccination and eradication programs. When used in a herd or flock vaccination program, inoculation of the Brucella melitensis Reverend 1 vaccine greatly decreases the prevalence of brucellosis in goats and sheep and therefore in human populations. Once the prevalence has been reduced, control of the disease may be achieved through the implementation of a program based on the combination of Brucella melitensis Reverend 1 vaccination of lambs, the serological testing of adults and the culling of serologically positive animals. Once the disease has been controlled by vaccination, eradication, i.e., the elimination of Brucella melitensis from the target animal population, can only be accomplished by a test and slaughter approach in flocks where the prevalence of the disease is very low. This requires the identification of animals and flocks, the control of animal movement, and adequate veterinary services and economic resources. In a test and slaughter program, 2 negative CFT results 6 months apart in all the animals in a flock is accepted as proof of eradication of the disease. The exclusive vaccination of replacement males and females can be effective in controlling the infection in countries with a low or moderate brucellosis prevalence. However, this exclusive vaccination of young replacement animals has failed to control brucellosis in some developed countries and is frequently not applicable in resource-limited countries because of the high prevalence of the disease and because in many regions the animals are kept under extensive management conditions that may be nomadic or semi-nomadic. As a result, the development of flock immunity is very low and unvaccinated adult females remain unprotected and may spread the infection further. Therefore, vaccination of the entire flock appears to be the only feasible method for controlling Brucella melitensis infection in small ruminants maintained under such extensive management conditions and high prevalence. In such difficult conditions, vaccination of both young and adult replacement animals every 2 years is the only technique suitable for reduction of the prevalence of the disease, and is therefore the first step towards successful control. Vaccination of pregnant with a full standard dose of Reverend 1 administered subcutaneously or conjunctival is often followed by abortion, particularly when they are vaccinated during early or midterm pregnancy. The induction of abortions when vaccinating pregnant animals means that there is no entirely safe strategy for vaccination with the Brucella melitensis Reverend 1 vaccine. Conjunctival vaccination is safer than subcutaneous vaccination but is not safe enough to be applied without taking the pregnancy status of the ewes or nanny goats into consideration, and it should be used only under restricted conditions. The number of abortions is, however, considerably reduced when adult female animals are vaccinated conjunctively just before mating or during lambing and lactation. Moreover, the vaccination of lactating animals with Reverend 1 is not followed by excretion of the vaccine strain in milk. Therefore, when mass vaccination is the only means of controlling the disease, a vaccination campaign should be recommended using the standard dose of Brucella melitensis Rev.1 administered by the conjunctival route applied when the animals are not pregnant. Production of Brucella or Brucella melitensis Rev.1 vaccine is based on a seedlot system. Seed cultures to be used in vaccines should originate from reference centers. They must conform to minimal standards for viability, smoothness, residual infectivity and immunogenicity. Guidelines for the production of vaccine are given in the WA Manual of Standards for Diagnostic and Vaccines. The Chinese live and smooth bee suis S2 vaccine that was apparently successfully used in field experiments in China and Libya did not provide adequate protection against Brucella melitensis infection when tested in controlled experiments. The Brucella abortus RB51, a rough attenuated strain, also did not protect sheep effectively against Brucella melitensis. Brucellosis in pigs is caused primarily by biovar 1, 2 or 3 of all 5 biovars of Brucellosis. Clinical signs in sows are abortion, birth of stillborn piglets, perinatal mortality, mastitis, arthritis and infertility. Orchitis and sterility may occur in infected boars. In endemic situations suis infections may be subclinical and farmers may be unaware that their herds are infected. Brucellosis biovars 1 and 3 are important zoonosis. Porcine brucellosis was first described by Traum in Indiana, USA, in 1914. The disease has a worldwide distribution, and despite the paucity of data its prevalence is considered to be low. Swine brucellosis appears to be endemic in most parts of South America and Southeast Asia, and is also believed to be widespread across sub-Saharan Africa. Although reported in Mozambique, not been diagnosed in South Africa. In the USA and Australia the infection, caused by suis biovar 1, is mainly restricted to feral pigs, while the wild boar, suis scrofa, and the European hare, Lepus europaeus, are the wild reservoirs of swine brucellosis caused by suis biovar 2 in continental Europe, except Finland, Norway, and Sweden. Brucellosis organisms are small, gram-negative, coccobacilli occurring naturally in the smooth S form and whose colonies cannot be differentiated macroscopically from isolates of other S. Brucella species. There are 5 recognized biovars of Brucellosis. Biovars 1, 2 and 3 are main causes of brucellosis in swine. Biovar 4 has been isolated from reindeer, in the Arctic including Russia, Alaska and Canada where it is considered to be endemic in semi-domesticated reindeer. Brucellosis has also been isolated sporadically in moose, American bison, muskoxen, Arctic foxes and wolves in the Arctic and constitutes a serious zoonotic problem. Brucellosis biovar 5 has been isolated exclusively from wild rodents in the former USSR. Very rarely brucellosis in pigs is caused by abortus and melitensis. Brucellosis biovars 1, 2 and 3 can be isolated in suitable culture media, and identified by simple bacteriological and molecular tests. Brucellosis biovars 1 and 3 are mainly pathogenic for pigs and humans but have also been reported to infect cattle sporadically. 6. These 2 biovars have a worldwide distribution. Epidemics of brucellosis in humans have been reported in the USA among packing house workers where the usual source was infected pigs, and also in Australia among those involved in the killing and slaughter of feral pigs. Human brucellosis caused by suis biovars 1 and 3 is thus almost entirely occupational, except in countries such as Brazil and Colombia and South America, where suis has become established in cattle, and brucellosis in humans caused by biovars 1 and 3 is emerging as an increasingly serious public health problem as a result of the consumption of unpasteurized milk because of infection of the bovine udder. Brucellosis biovar 2 differs from Brucellosis biovars 1 and 3 in that it is restricted to continental Europe and northern Africa and infects wild boar and the European hare, which are the wild reservoirs. Otherwise, swine brucellosis due to Brucellosis biovar 2 closely resembles the disease caused by Brucellosis biovars 1 and 3. Remarkably, Brucellosis biovar 2 is very rarely a human pathogen and has only been reported twice as the cause of human brucellosis in immune-compromised patients. Brucellosis biovar 2 can sporadically infect cattle. However, Brucellosis biovar 2 is not likely to be transmitted between cattle because they are spillover and not preferential hosts for Brucellosis biovar 2, and are thus not likely to sustain the infection. In modern industrial pig farming, infection is generally introduced into a herd by the purchase of infected animals. Boars may be the main source of infection, and transmission occurs chiefly via the semen, at mating. Indeed, persistent granulomatous lesions, in the testes and accessory sex organs, from which bacteria can be shed in the semen, for months or years, may be present in boars even though they appear healthy. Hence the potential exists for spreading the disease through artificial insemination. Other routes of spread are transplacental and the ingestion of aborted materials, infected milk or contaminated feedstuff. The feeding of contaminated kitchen waste, containing uncooked pork, may also be important. Transmission of suis biovar2 from wild boar to pigs in open-air breeding systems or backyard farms is thought to be mainly through the venereal route. Infected hares can be involved in suis biovar2 outbreaks in domestic pigs through swill feeding with offal from hunted infected hares or ingestion of dead hares. In suitable environmental conditions, Brucella suis can survive for several months. Animal premises may thus remain contaminated for prolonged periods. Direct sunlight considerably reduces the survival time. Brucella suis resists drying and freezing, and also survives for considerable periods in cold climates in aborted fetuses and manure. However, environmental bacterial persistence is accepted to be of low epidemiological importance, since direct or close animal-to-animal contact is required for transmission. Maintenance of Brucella suis in a population requires continued infection of susceptible hosts. The course of the clinical disease is usually acute when Brucella suis is introduced for first time into a non-infected herd of pigs. Infection spreads rapidly in a herd, and more than 50% of the animals may become infected in a relatively short period of time, manifesting mainly as a high rate of abortions and infertility. Infection rates of up to 70 to 80% are not uncommon in the early stages of outbreaks, resulting in severe economic losses. If no control measures are implemented, some infected individuals will either show a chronic form of the disease, with or without excretion of Brucella suis, or more infrequently free themselves of the infection, self-cure, while others may remain uninfected. The latter are at risk and may subsequently become infected because of the presence of Brucella suis in the herd. This sequence of events explains why a herd may be infected for years, but for long periods may be considered to be free of clinical disease. The pathogenesis of Brucella suis infection in pigs has not been elucidated fully. Brucellosis in pigs is usually a chronic infection that persists for life. The infected animals develop inflammatory responses in lymphoid tissues and organs rich in reticuloendothelial cells, like the spleen. Hosts mostly become infected through mucous membranes. Through licking, sniffing, or ingesting aborted fetuses and fetal membranes, or ingesting contaminated milk, Brucella suis comes into contact with the oral and nasal mucosae, tonsils, and gastrointestinal or reproductive mucosae. After entry, the pathogenesis of Brucella suis is believed to be similar to infection of preferential hosts by other Brucellae. Brucella suis localize initially in lymphoreticular tissues draining the site of infection, followed by a bacteremia that may persist for up to 90 days or more. During and subsequent to the bacteremia, localization of Brucella suis occurs in various tissues including the male and female genital organs, skeletal system, joints, mammary glands, lymph nodes, spleen, liver, kidneys, and urinary bladder. Localization of Brucella suis in these organs or tissues accounts for the main clinical signs of the disease, and the main routes of excretion. Infection with Brucella suis may also be subclinical. Like Brucella infections in other animal species, the onset of infection depends on the virulence and dose of the infecting strain as well as the susceptibility of the host as determined by innate and acquired immunity mechanisms. It has been reported that the virulence of Brucella suis biovars 1, 2 and 3 are similar in pigs, but no experimental evidence is available. The clinical signs of brucellosis in pigs, when present, vary but are similar to those in cattle, sheep and goats. Reproductive failure characterized by abortion, stillbirth and infertility in sows and orchitis, epididymitis and infertility in boars are the main clinical features of Brucella suis infection. Abortion may occur at any stage of pregnancy. Early abortion may be missed, and one may think that there is an infertility problem in the herd. Sows usually do not abort more than once following infection but may become infertile thereafter. Some piglets infected in utero may die within a few hours of birth, the mortality rate often being very high, but others survive and retain the infection into adulthood. Sows may suffer from mastitis. In boars, there may be no clinical signs or there may be swelling of the testes, which subsequently undergo atrophy. Mortality in older pigs is usually low, but chronic disease causes economic losses because of the forced culling of boars and sows as a result of infertility caused by lesions in the genital tract or as a result of lameness caused by arthritis, bursitis and tenovaginitis. The majority of visible lesions in the genital organs consist of progressively enlarging granulomas that may become calcified. In the uterus and fallopian tube, they are generally 2 to 5 mm in diameter and have a miliary distribution. In addition to this miliary granulomatous metritis, mucoid endometritis may be present, in which numerous Brucella organisms are present in the exudate in the lumen of the uterus. In boars, it is often possible to isolate the organisms in the absence of gross pathology. However, orchitis and infection of the accessory sex glands are common. Orchitis caused by Brucellosis results in multiple abscesses accompanied occasionally by fibrinopurulent or hemorrhagic periorchitis. Testes may appear enlarged or atrophic. Abscesses in the testes or epididymis have a caseous core surrounded by epithelioid macrophages with a connective tissue capsule. Infection of accessory glands can be associated with vesicular gland hypertrophy, and microabscesses in vesicular glands, prostate, or bulbourethral glands. Calcified foci may also be seen in the testes and accessory sexual glands and organs, particularly in the epididymis and seminal vesicles. Testicular atrophy and a variable degree of enlargement of the epididymis are characteristics of the chronic phase of the disease. The inflammatory reaction, which may be present in one or more joints, is fibrinopurulent and may cause lameness. If the vertebrae, often the lumbar region, are involved, granulomatous osteomyelitis develops that can destroy adjacent intervertebral discs. Such affected animals often manifest with posterior paresis and sometimes paralysis. No specific lesions occur in aborted fetuses. Rangifer brucellosis in reindeer and caribou is caused by Brucellosis biovar 4 throughout the Arctic region, Siberia, Canada and Alaska, and constitutes a serious zoonotic problem. When clinical signs are present in these animals, abortions and metritis are seen in females and orchitis occurs in males. In both sexes, abscess formation in joints, bursitis and lameness are observed in some cases. Transmission to humans may be by direct contact, or through the consumption of raw milk or other inadequately heated products from infected animals. Diagnosis: Brucellosis infection in pigs may be present in the absence of typical clinical signs or gross pathology, which is not pathognomonic. However, in endemic regions, organs, particularly the genital organs, showing granulomatous lesions during routine postmortem inspection, can be suggestive of infection. Direct tests. A presumptive diagnosis can be made by microscopic examination of stamp-stained smears from vaginal swabs, placentas and/or aborted fetuses, and semen. However, this test lacks sensitivity and specificity, and the isolation of Brucellosis on appropriate culture media allows the only accurate diagnosis. Vaginal secretions, swabs, from aborted sows, milk, semen, fetal membranes and samples from aborted fetuses, stomach contents, spleen and lung, are the samples of choice in live pigs. At necropsy, the spleen and lymph nodes from the head, mammary, and genital tract are the samples of choice. Brucellosis can also be isolated from the testis, epididymis, vesicular glands, prostate and bulbourethral glands of boars. Brucellosis grows well at 37 degrees Celsius on general multipurpose culture media, i.e., blood agar, without the addition of blood or serum, can grow in air, but the highest sensitivity of the culture is obtained in a 5 to 10% CO2 atmosphere. The simultaneous use of both ferrules and the seed as media is recommended for the isolation of smooth Brucellae, including Brucellosis. Colonies generally appear after 3 to 4 days, but cultures should not be discarded as negative until after 8 days of incubation. Brucellosis colonies are morphologically indistinguishable from other smooth Brucella and can be presumptively identified by positive reactions in the oxidase and urease tests and by agglutination with monospecific antisera. With DNA from the isolated colonies, several PCR procedures allow proper identification of Brucellosis. However, due to the presence of DNA inhibitors in animal tissues and field samples, the sensitivity of these methods is significantly lower than that of classical microbiological culture. Indirect tests. Virtually all serological tests used for the diagnosis of brucellosis in ruminants have been applied for diagnostic purposes for brucellosis in pigs. None of the conventional serological tests used in domestic ruminants are fully reliable for diagnosing pig brucellosis when applied to individual animals. Therefore, it is recommended that serological tests be interpreted on a herd rather than on an individual animal basis. The serological tests for brucellosis in pigs are performed using the same cell wall-associated B. abortus smooth lipopolysaccharide, SLPs, antigen used for the diagnosis of B. abortus and B. melitensis infections in domestic ruminants, CB. melitensis and B. abortus. It must be noted, however, that Brucellosis may be isolated from seronegative pigs, which has important implications in eradication programs. It has been shown that natural and experimentally induced Y. enterocolitica infections produce false positive serological reactions, FPSR, in SLP's brucellosis tests that are virtually indistinguishable from those caused by brucellosis. Accordingly, none of the above SLP's tests provide 100% specificity when testing a brucellosis-free pig population affected by the FPSR. This problem is particularly important and frequent in brucellosis-free countries and interferes with the international trade of live pigs. Several serological tests have been proposed to solve this FPSR problem in pigs, including both C. elisa and I. elisa with SLPs, as well as an I. elisa using an extractive rough, R. Brucell. The results of these investigations show that serial and cumbersome testing schedules would be necessary to increase the specificity of the diagnosis, and moreover, none of them has been proven to be fully specific to differentiate brucellosis from FPSR. Indirect tests. Soluble cytosolic proteins are presumed to be specific from the Brucella genus, thus being potential candidates to resolve the FPSR problem. When used in serological tests, these antigens do not demonstrate suitable diagnostic performance in pigs. Lymphocyte transformation tests with these proteins have been studied to measure cell-mediated responses of infected pigs. Preliminary results suggest that there is good correlation of the results of these tests with those obtained by bacteriological isolation of Brucellosis from tissues. None of these lymphocyte tests are in common use and proven effective to resolve the FPSR problem. Allergic skin tests have been widely used in Eastern Europe, the former USSR and China for the diagnosis of brucellosis in pig herds. As the antigens used for the allergic tests are the Brucella cytosolic proteins, no cross-reactions with Y. enterocolitica are expected, and thus the diagnostic specificity is very high. However, the sensitivity of the allergic tests is similar to that of the serological tests in that they are not reliable for the diagnosis of the disease in individual pigs, but they are very valuable when interpreted at herd level. A commercially produced allergen is available for the allergic skin test has been shown to be very useful in the context of FPSR in cattle control. Swine brucellosis is usually not a disease that occurs on intensive pig farms with modern indoor facilities. It is critical to prevent Brucellosis infected animals from entering these intensive systems, particularly nucleus breeding herds at the top of the pyramid and artificial insemination centers. Thus, strict biosafety measures must be implemented in these intensive farming systems as well as where domestic pigs are reared in outdoor breeding systems or small backyard farms to avoid contact with feral pigs or wild reservoirs. Where there is a risk of introducing swine brucellosis, regular serological surveillance should be performed on a herd basis, using the skin test as a confirmatory test at herd level whenever positive serological reactions are detected. Adequate fencing, although expensive, to avoid contact with wild reservoir hosts of Brucellosis is important to prevent infection of free-range pig farms. Eradication of Brucellosis from a herd is based solely on the depopulation of all animals in the infected holdings. However, culling of large herds kept in or outdoor farming systems may not be economically feasible or impracticable. An alternative control strategy could be testing with culling of infected animals. However, the current serological tests are not suitable to apply at an individual level and should be used at a herd level. It is therefore not possible to eradicate the disease from an infected herd by means of serological test and removal programs. Alternatively, Test and culling strategies based on the use of a combination of serological tests and the skin test in breeding stock can be used to eradicate the disease in a herd, as some infected animals that are negative serologically react positively to the skin test, and vice versa. Where practicable, it is thus useful to perform a combination of both serological and skin tests. When total depopulation is not feasible and eradication not possible, antibiotic treatment, alone or in combination with test and culling, can be applied to control the disease, minimizing the clinical effects and economic impact. Although further work is necessary, a combined regimen of oxytetracycline and tildapyrosin can be a cost-effective practical alternative to control swine brucellosis where depopulation is not feasible. In addition to antibiotic treatment, selective culling, i.e., intensifying the slaughter of sows that aborted or that are infertile, and testing and slaughter infected pigs on the basis of skin test results, could significantly reduce the prevalence of disease in a herd. Replacement stock should originate from herds known to be officially free of brucellosis. According to the Office International of Epizooties, for international and other trade purposes, the disease status of the herd and that of the area in which the herd is situated are of more importance than the testing of individual animals. No vaccine is effective in preventing brucellosis in swine. Infectious epididymitis. Brucella ovis infects primarily sheep and is one of the most common causes of epididymitis in rams and a rare cause of abortion in ewes and neonatal mortality in lambs. Low reproductive rates may occur in affected flocks. Similar clinical manifestations have been occasionally reported in male red deer, Cervus alipus, in New Zealand. Is not a zoonotic disease. Brucella-like organisms, which were first isolated from the lesions of rams with epididymitis and from ewes in Australia and New Zealand in 1953, were assigned to a new species, Brucella ovis. In Africa, rams with clinical or subclinical epididymitis and low fertility as a result of Brucella ovis infection were diagnosed for the first time in 1956. With few exceptions, Brucella ovis infections occur practically in all sheep-raising countries. In Australia, it has been reported that up to 80% of young rams in certain studs or flocks are infected. In Europe, Brucella ovis infections have been described mainly in southern European countries and more recently also in Eastern Europe. The infection is rare in Northern America, whereas Brucella ovis infection is one of the major causes of low fertility or infertility in rams in Argentina and Brazil. The infection is widespread in Africa. Brucella ovis is similar to the other Brucella spp. In its morphology, staining properties and cultural characteristics can be differentiated on the basis of its colonial morphology, as it is always isolated in the rough phase, negative reactions in the oxidase and urease tests, lack of agglutination with specific A and M antisera, and susceptibility to the RC phage. Culture of the organism requires an atmosphere containing 5 to 10% carbon dioxide, and enrichment of the culture media with 5 to 10% blood or serum. Optimum growth occurs at 37 degrees Celsius while no growth occurs at temperatures below 20 degrees Celsius or above 40 degrees Celsius. On primary culture after 3 to 4 days of incubation on media such as blood agar, the colonies are grayish-white, up to 0,5 to 2, 5 mm in diameter, non-hemolytic, circular, convex, rough phase, glistening, translucent and entire-edged. Epidemiology. Sheep are naturally infected by B. ovis. Besides experimental infection, is serological evidence of natural exposure in goats. Red deer are also susceptible and transmission occurs through direct contact with infected rams. White-tailed deer are susceptible to experimental infection. The disease is spread primarily by infected rams. Brucella ovis may be excreted in the semen of infected rams even before the development of lesions. Clinically or subclinical infected rams may excrete Brucella ovis in their semen for years. Infection may spread from ram to ram as a result of sexual activity or venereal during coitus when uninfected rams mate with ewes that passively harbor the bacteria. Ewes generally only transfer organisms mechanically from infected to uninfected rams when they are mated in succession by different rams during the same heat period. Vasectomized and cryptorchid rams are equally vulnerable as a result of direct transmission through contact. homosexual activity and venereal spread through ewes. Direct ram-to-ram and venereal transmission result in a higher prevalence of infection in flock rams than in stud rams. This is probably due to the differences in mating and management practices between these two categories of rams, such as the regular examination of breeding rams and new introductions, individual versus mass mating, and the more isolated rearing and housing of stud rams. High prevalence and even epidemics have been reported in non-breeding rams housed together. Most ram-to-ram infections occur via the oral route. Housed rams establish hierarchies, head-to-head combats, and it is frequent that dominated rams, after being mated by the dominant rams, lick the prepuce of these dominant rams as an act of submission. If these dominant rams are infected, the probability of harboring Brucella ovis in the prepuce is very high and hence transmission may occur. Natural infections of ewes usually persist for 10 to 14 days in uterine discharges of ewes that have aborted. It is, however, believed that ewes do not play a relevant role in the maintenance of Brucella ovis in infected flocks, and accordingly, eradication schemes are based exclusively on the serological testing and culling of seropositive rams. However, field and experimental data support the view that ewes should not be disregarded as a source of infection for rams, and particularly of potential congenital infections in lambs. It has, in fact, been reported that ewes could have played a role in maintaining Brucella ovis infection in flocks despite the testing and culling of seropositive rams. Although Brucella ovis has a relatively low ability to induce abortion in pregnant sheep and is seldom transmitted to lambs, can cause persistent mammary infection in ewes. These persistently infected ewes during at least 2 successive lactations will play a role in the epidemiology of the infection. Moreover, Brucella ovis can be excreted by the vaginal route during estrus by a significant proportion of infected ewes, allowing the possibility of ewe-to-ram transmission at mating. Pathogenesis. Rams become infected after penetration by Brucella ovis of the mucous membranes of the prepuce, penis, nasal cavity or conjunctiva. This is followed by bacteremia and localization of the infection in lymph nodes and organs such as the epididymis, ampullae, seminal vesicles, bulbourethral glands, spleen, liver and kidneys. Organisms are first excreted in the semen from between 31 and 45 days following exposure. Gross lesions only develop in the genitalia. In rams, the earliest lesions usually occur in the tail of the epididymis, but lesions in the head and/or body of the epididymis may develop later. Initially, the bacteria cause degeneration and necrosis of the epididymal epithelium, resulting in leakage of semen into the interstitial tissues, where it provokes a severe inflammatory reaction and the development of spermatic granulomas. Similar inflammatory changes may also occur in the vas deferens, ampullae, seminal vesicles, bulbourethral glands and testes. Inflammatory cells, particularly neutrophils, in the semen, and a decrease in the production and quality of sperm as a consequence of testicular degeneration all lead to lower fertility or infertility. Infertility may also be caused by the total cessation of spermatogenesis or by obstruction of the epididymis by spermatic granulomas and the development of a spermatocele. There is a direct relationship between the semen quality, extent of the epididymal lesions and the number of leukocytes present in the semen. The reduction in fertility is caused to both low spermatozoa counts and the high number of defective spermatozoa, defects of the spermatozoid's head and neck being common. In ewes, organisms enter mainly through the vaginal mucosa. In pregnant ewes, the ensuing bacteremia causes placentitis that may result in abortion or the birth of lambs with reduced birth weights. However, despite induction of severe endometritis, Brucella ovis has a relatively low capacity to induce abortion in sheep. After experimental infection, the uterus and the iliac and supramammary lymph nodes are the main target organs of Brucella ovis infection in ewes. A detectable humoral immune response in rams initially appears 1 to 2 weeks after exposure, but uniformly positive titers are only obtained from the 4th week onwards in the complement fixation test. Clinical signs. The interval between the infection and the development of lesions in rams varies considerably, from 50 to 250 days. In rams, the first detectable abnormality may be a marked deterioration in semen quality associated with the presence of inflammatory cells and organisms. The most consistent clinical sign is enlargement, particularly of the tail of the epididymis figure 1, which may be barely perceptible or up to a 4 or 5-fold increase in size. The head, body or the entire epididymis are less often affected. The lesions often occur unilaterally, but bilateral involvement is relatively common. Rams suffering from acute epididymitis are not usually systemically affected. The entire testis on the affected side may be hot, swollen and edematous, but only localized swelling of the epididymis is detectable in animals that are less severely affected. Clinically detectable lesions may be acute to chronic. Although chronic lesions may follow an acute epididymitis, they more commonly develop insidiously without clinical evidence of the acute phase. Chronic epididymitis is clinically characterized by enlargement and an increase in consistency of the affected parts. As a result of fibrous adhesions, the mobility of the affected testis in the scrotum is often reduced. The marked increase in scrotal circumference caused by the epididymal and testicular lesions can be seen from a distance. Ultrasonography is useful for clinical assessment in these cases. The testis is seldom primarily affected. In some cases, the testis on the affected side may be slightly atrophied and have a somewhat softer consistency than normal, while in others with severe, chronic epididymitis, the testis may be severely atrophied and firm. Affected rams may be sterile, or have reduced fertility. The degree of impairment depends on whether the lesions are uni or bilateral, and on the course and severity of the lesions. The libido of affected rams remains unaffected. Ewes abort very rarely as a result of Brucella ovis infection. In experimentally infected pregnant ewes, abortions may affect from none to about 30% of the inoculated animals. However, field reports suggest that as many as 50% of the pregnant ewes may abort. Figure 1. Asymmetry and enlargement of the tail of the epididymis. Typical lesions in the affected epididymis include solitary or multiple spermatocells and spermatic granulomas, which contain a creamy fluid or inspissated, caseous material. Figure 2. Thickening due to the presence of a low-grade, non-purulent inflammatory response. Figures 3 and 4, and fibrosis of the interductal connective tissue and the tunica albuginea. In most cases, fibrous adhesions form between the tail of the epididymis, the parietal tunica vaginalis, and the distal pole of the testis. Testicular degeneration and atrophy, which is usually more severe in rams with widespread and severe adhesions, may accompany the epididymal lesions. In most cases, however, the changes in the testis are minimal and nonspecific. Changes in the vas deferens and accessory sex glands may be similar to those in the epididymis. Semen quality is determined by the extent and severity of the lesions in the epididymis, testes, and accessory glands. Poor semen quality is characterized by reduced density of the ejaculate, due to decreased numbers of spermatozoa, reduced motility and longevity of spermatozoa, an increase in the proportion of abnormal spermatozoa such as those with detached sperm heads, mid-piece abnormalities, bent tails, coiled tails, and tails tightly coiled around the heads of spermatozoa, and the presence, in many cases, of varying numbers of leukocytes, particularly neutrophils. Figure 2. Spermatic granuloma of the tail of the epididymis in a case of Brucella ovis infection. The carcasses of aborted lambs are not autolyzed but are dehydrated, and they manifest fibrinous peritonitis. The accompanying placentitis is characterized by a yellowish fibrinous exudate that is present particularly in the intercotyledonary areas. Histologically, the lesions in the placentas are characterized by multifocal suppurative inflammation. Fetal lung tissues manifest suppurative bronchitis, bronchiolitis, and bronchopneumonia. Figure 3. Chronic lesions, degenerative changes in the epididymal duct. Figure 4. Chronic lesions, spermatic granuloma in the epididymis. The diagnosis of Brucella ovis infection in sheep includes palpation of the external genitalia of the rams, microscopic examination of stained semen smears, bacteriological culture of semen and exudate from lesions, and serology. Molecular methods are available, including a Brucella ovis-specific PCR protocol. The presence of Brucella ovis and neutrophils in semen smears stained by the stamp method, particularly rams that are subclinically affected, can be useful for the diagnosis of the disease. Figure 5. However, the presence of bacteria causing epididymitis with similar morphology and stamp staining characteristics can lead to misdiagnosis. Therefore, immunofluorescence techniques have been developed. Although there is usually a good correlation between the presence of neutrophils and Brucella ovis organisms in semen smears, the presence of neutrophils alone is not a confirmatory diagnostic. Figure 5, semen smear stained by the stamp method. Note the numerous acid-fast bacteria. Brucella ovis may be excreted in semen by only some infected rams, and this excretion can be intermittent, limiting the sensitivity of the semen culture for the diagnosis of infection. In ewes that have aborted, Brucella ovis infection can be diagnosed by demonstration of the organisms in smears made from uterine exudate and by bacteriological culture of uterine exudates and milk. In aborted fetuses, bacteriological cultures of the abomasal content, spleen, lungs and liver should be made in order to isolate the organism. Several serological tests can be applied to the diagnosis of Brucella ovis. The most efficient and widely used tests are the agar gel immunodiffusion, AGID, complement fixation test, CFT, and the indirect enzyme-linked immunosorbent assay, ELISA. Several countries have adopted various standard diagnostic techniques for B. ovis, but for international trade, the only test prescribed by the WA and the European Union is the CFT. However, it has been demonstrated that the AGID test shows similar sensitivity to the CFT, and that it is easier to perform and is very practical for routine diagnosis in non-specialized laboratories. Numerous independent studies have shown that the ELISA is more sensitive and specific than either the CFT or AGID test, but to date, no standardization of such tests exists. Most of the serological tests for the diagnosis of smooth colony Brucella spp. infections, B. melitensis, B. abortus and B. suis, have been developed to detect antibodies directed against antigens associated with the smooth lipopolysaccharide that is absent in the naturally rough Brucella ovis. Although false negative and false positive results occur with the CFT, no false positives have been encountered in flocks known to be free of the disease, but they do occur in infected flocks. Moreover, the CFT has other important disadvantages such as complexity, obligatory serum inactivation, anticomplementary activity of some sera, difficulty to perform with hemolyzed sera, and prozone phenomena. Brucella ovis is mainly detected by PCR or bacteriology in sexual organs and regional lymph nodes, and only rarely at systemic sites of infection. The Brucella ovis specific PCR assay is suitable for routine diagnosis of this disease, allowing a concurrent differential diagnosis with Brucella melitensis, and it can be used as an alternative to bacterial isolation. The impact of bacterial infections of the genital system of rams can be extremely important, particularly if infected rams are not properly diagnosed in time, and are allowed to enter into the mating period. The clinical examination of external genitalia is largely nonspecific because there are many other causes of epididymitis. Epididymitis in rams as a result of Brucella ovis should be differentiated from similar lesions caused by Actinobacillus seminis, Actinobacillus actinomycetum comitans, other Actinobacillus spp, Histophilus somni, Corynebacterium pseudotuberculosis, Chlamydia spp, and other Haemophilus spp. As recently reviewed, nodular enlargement of the epididymis and testes detected clinically may also be caused by trauma, paradidymal cysts, Cysticercus tenuicollis cysts, Sertoli cell tumors and seminomas, and by spermatocells and spermatic granulomas resulting from non-infectious causes. Nodular enlargement within the scrotum due to varicocele should also be considered. Control is based on the elimination of infected animals, vaccination, or a combination of the two approaches. Control programs are aimed at preventing infection from taking place as well as eliminating infected rams. Management procedures such as the rearing and weaning of male replacement lambs in isolation from other rams, particularly breeding rams, timely weaning of ram lambs, early replacement of older rams, avoidance of the use of susceptible young rams in multiple sire breeding systems, testing of rams before the mating season, and introduction only of tested and/or vaccinated rams, are important in the control of the disease. Brucellosis due to Brucella ovis should not be considered as an exclusive infection of rams and could be present in a certain proportion of ewes in some infected flocks. Although the precise mechanism of transmission has not been established, the maintenance of infection in some flocks submitted to exclusive testing and culling of infected rams can be due to the existence of infection in ewes. Accordingly, in these problem flocks, ewes should be included in any Brucella ovis control program. Vaccination with the Reverend 1 mutant strain of Brucella melitensis has proved to be highly effective in preventing Brucella ovis infection. The induced immunity against Brucella ovis infection lasts for up to 4 years. In areas where Brucella melitensis infections are known to exist, the use of the Reverend 1 vaccine by the conjunctival route, as opposed to the subcutaneous route, may be advisable in rams, as the conjunctival inoculation results in similar immunity while minimizing the post-vaccinal serological response in serological tests for Brucella melitensis, thus reducing the serological interference in diagnostic tests. The main problem of Reverend 1 is that its use are prohibited in countries free from Brucella melitensis, and other vaccines, if available, should be used. However, no other vaccine than the Reverend 1 is recommended by the WHO. The live smooth Brucellosis strain 2, Chinese vaccine, and the live rough B. Abortus RB51 vaccine do not confer adequate protection against Brucella ovis in rams. Some degree of efficacy against Brucella ovis has been shown using inactivated vaccines. A vaccine containing aluminum hydroxide adsorbed Brucella ovis bacteria probably only protects rams from the clinical disease and does not prevent the establishment of a carrier state. Another inactivated vaccine in special adjuvants has been shown to be as effective as Reverend 1 against Brucella ovis in rams. In the European Union, no compulsory surveillance for the disease is done in flocks. Neither an eradication program nor a compensation scheme for culling animals in infected flocks is foreseen. The treatment of Brucella ovis infected rams with long-acting oxytetracycline and dihydrostreptomycin results in a reduction in the shedding of the pathogen in the semen, but it is expensive and conclusive elimination of the infection from all rams has not been proven. However, addition of antibiotics to cryopreserved semen inactivates Brucella ovis, and therefore it may be a suitable control measure where artificial insemination is employed. Glanders and melioidosis. Glanders is a contagious and fatal disease of perissodactyls or odd-toed ungulates, horses, donkeys, and mules, caused by infection with the bacterium Burkholderia mallei. The pathogen causes nodules and ulcerations in the upper respiratory tract and lungs. A skin form also occurs, known as farcy. Melioidosis is an infectious disease caused by Burkholderia pseudomallei in humans and animals, and sometimes resembles glanders in horses. Burkholderia mallei has evolved from Burkholderia pseudomallei by reduction of genetic information and is phylogenetically considered as a clone, a pathovar of B. pseudomallei. Control of glanders and melioidosis requires testing of suspect clinical cases, screening of apparently normal equids, and elimination of reactors. Stable hygiene and manure management are imperative. Burkholderia mallei and Burkholderia pseudomallei can be transmitted to humans, all infected or contaminated. Material must be handled in a laboratory with appropriate biosafety and biosecurity controls following a biorisk analysis. Felidae and other species are susceptible, and infections are usually fatal. Importantly, this organism is zoonotic, with a 95% fatality rate in untreated septicemia cases, and is therefore considered a potential bioterrorism agent. Glanders is one of the oldest diseases known and once was prevalent worldwide. It has now been eradicated or effectively controlled in many countries, including the USA. In recent years, the disease has been reported in the Middle East, Pakistan, India, Mongolia, China, South America, Africa, and some European countries, including Russia. Burkholderia mallei organisms are fairly numerous in smears from fresh lesions, but scarce in older lesions. Is aerobic and facultative. Smear should be stained with methylene blue or gram stain. Growing well at 37 degrees Celsius. The gram-negative rods have rounded ends. It grows well, are 2 to 5 microns long and 0.3 to 0.8 microns wide, with granular inclusions of various size. Of cultures is recommended. The bacteria are generally located extracellularly and frequently stain irregularly. The tiny grayish shiny colonies of bees used. Mallei on sheep blood agar readily visible capsule under the light microscope and do not form spores. Careful observation is needed not to overlook the presence of a capsule-like cover has been verified by electron microscopy. After a few days on glycerol composed of neutral carbohydrates and serves to protect the cell from unfavorable environmental factors, growth can be observed. Unlike other organisms in the Pseudomonas group and its close relative, the growth thickens and becomes dark brown and tough. Burkholderia malleae has no flagella and is therefore potato agar, and in glycerol non-motility is the most slimy phenotypic characteristic diagnostically and must be demonstrated. The growth is much less effusive, and growth is poor. The organisms are difficult to detect in various commercially available where they may have a beta-selective agars enable the growth in culture media. They vary in appearance depending on the age of the culture and obtained under sterile conditions. In older cultures, malleae is often overgrown by other bacteria, which makes isolation difficult. Form on the surface various of biochemical characteristics can be used for the differentiation of B. malleae from B. Pseudomalleae, as B. malleae has lost many fermentative activities during phylogeny. Commercially available laboratory biochemical identification systems can be used for confirmation that an organism belongs to the Pseudomonas group. In general, however, commercially available systems are not suitable for unambiguous identification of members of the steadily growing number of species within the genus Burkholderia. Lack of motility is therefore of special relevance. Cell matrix-assisted laser desorption, ionization mass spectrometric typing can be used. A bacteriophage specific for B. malleae is available. Reference laboratories or specialized laboratories use B. Pseudomalleae specific antibodies that are not commercially available and that have not been validated for use in equids. No validated procedures for the isolation of B. Pseudomalleae from horse samples exist. Cultivation may be successful from samples of ulcers, lesions or excretions. Standard media, blood or McConkey agar, are used for isolation of B. Pseudomalleae and selective enrichment techniques as Ashdown agar, Galliman's broth or B. Pseudomalleae selective agar, BPSA, have been developed. Colony morphology varies from smooth to rough depending on the medium and strain, and often becomes wrinkled after a few days of incubation. A metallic sheen over the area of confluent growth on Columbia agar is an important feature for the presumptive identification of B. Pseudomalleae. Outside the body, B. malleae shows little resistance to drying, heat, light or chemicals, so that survival beyond 2 weeks is unlikely. Under favorable conditions, however, it can probably survive a few months. Burkholderia malleae can remain viable in tap water for at least 1 month. For disinfection, benzalkonium chloride, sodium hypochlorite, iodine, mercuric chloride and alcohol, and potassium permanganate have been shown to be highly effective. Phenolic disinfectants are less effective. Burkholderia pseudomalleae can survive in water 16 years, in muddy water for up to 7 months and in soil in the laboratory for up to 30 months. Chlorine has only a bacteriostatic effect on the agent as bacteria were recovered from water containing up to 1,000 parts per million. Free chlorine. Epidemiology. Contagious disease in equids and outbreaks may also occur in felids living in the wild or in zoological gardens. Susceptibility to glanders has been proven in camels, bears, wolves and dogs. Carnivores may become infected by eating infected meat, but cattle and pigs are resistant. Small ruminants may be infected if kept in close contact with glanderous horses. Glanders is transmissible to humans by direct contact with diseased animals or with infected or contaminated material. In the untreated acute disease, the mortality rate can reach 95% within 3 weeks. However, survival is possible if the infected person is treated early and aggressively with multiple systemic antibiotic therapies. A chronic form with abscessation can occur. Glanders has been eradicated from many countries by statutory testing, culling of infected animals, and import restrictions. It persists in numerous Asian, African and South American countries and can be considered a re-emerging disease. Glanders can be introduced into glanders-free areas by movement of equids. Burkholderia pseudomalleae is the causative agent of melioidosis and originates from Southeast Asia. It is an important soil bacterium, denitrifying organic materials and is ubiquitous in those areas. Thus, B. pseudomalleae is stable in the environment. It is also naturally resistant to various antibiotics and disinfectants. Burkholderia pseudomallei has an extremely broad host range including wildlife, farm animals and humans. Glanders is commonly contracted by ingestion of food and water contaminated with nasal discharges of carrier animals, contact with contaminated harness components, ingestion of meat from affected horses. Clinical. After an incubation period of 3 days to 2 weeks, in acute form of glanders usually we identify septicemia, high fever, weight loss, a thick, mucopurulent, yellowish nasal discharge. Respiratory signs. Death occurs within a few days. The chronic form is common in horses and is seen as a debilitating condition with nodular or ulcerative lesions of the skin and internal nares. Infected animals may live for years and continue to disseminate the organism. In some, the infection may be latent and persist for long periods. Donkeys and mules are more severely affected than horses. Lesions. Nasal, pulmonary, and cutaneous forms of glanders are recognized, and an animal may be affected by more than one form at a time. In the nasal form of glanders, nodules develop in the mucosa of the nasal septum and lower parts of the turbinates. The nodules degenerate into deep ulcers with raised irregular borders. Characteristic star-shaped cicatrices remain after the ulcers heal. In the early stage, the submaxillary lymph nodes are enlarged and edematous and later become adherent to the skin or deeper tissues. In the pulmonary form of glanders, small, tubercle-like nodules, which have caseous or calcified centra surrounded by inflammatory zones, are found in the lungs. If the disease process is extensive, consolidation of the lung tissue and pneumonia may be present. The nodules tend to break down and may discharge their contents into the bronchioles, resulting in extension of the infection to the upper respiratory tract. In the cutaneous form of glanders, Farsi, nodules appear along the course of the lymph vessels, particularly of the extremities. These nodules degenerate and form ulcers that discharge a highly infectious, sticky pus. The liver and spleen also may show typical nodular lesions. Histologically, there may be vasculitis, thrombosis, and infiltration of degenerating inflammatory cells. Diagnosis are suspected based on cutaneous nodules, exuding honey-like discharge, or nasal discharge with ulcers, on nasal mucosa. The typical nodules, ulcers, scar formation, and debilitated condition may provide sufficient evidence for a clinical diagnosis of glanders. However, because these signs usually do not develop until the disease is well advanced, specific diagnostic tests should be used as early as possible. Confirmed by culture, complement fixation test, and PCR. Culture of B. malei from lesions confirms the diagnosis. A test for delayed hypersensitivity is performed by intrapalpebral inoculation of malin extract, a secreted glycoprotein of B. malei found in culture supernatant. Infected hypersensitive horses develop a purulent conjunctivitis within 24 hours and swelling of the eyelid. The malin purified protein derivative, PPD, which is available commercially, is a solution of water-soluble protein fractions of heat-treated B. malei. The test is not generally recommended because of animal welfare concerns, however it can be useful in remote endemic areas where sample transport or proper cooling of samples is not possible. It depends on infected horses being hypersensitive to malin. Advanced clinical cases in horses and acute cases in donkeys and mules may give inconclusive results requiring additional diagnostic methods. The intradermopalpebral test is the most sensitive, reliable and specific malin test for detecting infected perissodactyls or odd-toed ungulates, and has largely displaced other methods. 0.1 ml of concentrated malin PPD is injected intradermally into the lower eyelid and the test is read at 24 and 48 hours. A positive reaction is characterized by marked edematous swelling of the eyelid, and there may be a purulent discharge from the inner canthus or conjunctiva. This is usually accompanied by a rise in temperature. With a negative response, there is usually no reaction or only a little swelling of the lower lid. Complement fixation CF is also used to screen for infection. Competitive ELISA is more sensitive than complement fixation and may become positive as early as 3 days after infection. PCR based on 16S and 23S RNA gene sequences may be used for specific identification. Prevention and treatment. Detection and elimination of confirmed cases. Treatment is contraindicated. There is no vaccine for glanders. Protective immunity involves T-cell responses elicited by live-attenuated bacteria. Prevention and control of glanders depend on early detection and elimination of affected animals, as well as complete quarantine and rigorous disinfection of the area involved. Successful control is dependent on efforts to establish awareness among horse and donkey owners in remote areas, in addition to provision of funds to cull affected animals. Treatment of glanders does not reliably produce a bacteriologic cure. Doxycycline, ceftriaxone, gentamicin, streptomycin, and combinations of sulfazine or sulfamonomethoxine with trimethoprim were effective in the prevention and treatment of experimental glanders.